Good morning, everyone, to the day 2 of our symposium of molecular imaging and stem cells. And in the day 1, we beautifully got an overview of how imaging is a part and parcel of carrying out our day-to-day research, be it identifying that whether the colony I'm picking, the colony I'm passaging, they are pluripotent stem cells or not, and what are the set and battery of markers and localization of those markers or the proteins needed to be checked when we are assuming and assigning that this cluster of cells are pluripotent stem cells, or these cells in our petri dish are mesenchymal stromal cells. Further, we also got an overview with respect to the sensors, optical sensors being used in vivo and in vitro. Beautifully, so many cellular events, be it from intracellular calcium to mitochondrial dynamics to migration to just docking, homing, and biodistribution. So we can understand through this presentations from, be it from industry and from academia, that imaging is woven with the stem cell field, and it definitely plays an important role to bring about advancement in this regenerative medicine field, and which is a good news for all of us. And it stems from our belief that seeing is believing. So with that, we will start the second day of the symposium. I'm happy and I thank all the participants who have joined in on time so that we can keep to our schedule. So we will welcome our first speaker of the day, Dr. Thomas Durkin. So a little brief bio about him. So Dr. Thomas is an assistant professor in the Department of Neurology and Neurosurgery at McGill University and is a member of the Center for Neurodegenerative Disease Group at the MNI. So Dr. Durkin received his Bachelor of Science from University College Dublin before moving to the USA to obtain his PhD in cell and molecular biology from the University of Notre Dame in 2007. For his postdoctoral research, Dr. Durkin joined the Parkinson's Research Group of Dr. Edward Fon, where he focused on the function of deubiquitinating enzymes in Parkinson's disease and other neurodegenerative disorders. His current research program is focused on the use of iPSCs and mouse models to understand how specific pathways are affected in Parkinson's and other neurodegenerative disorders. In addition to this, his academic appointment, Dr. Durkin is Associate Director for the Neuro's Early Drug Discovery Unit, EDDU, focused on the use of iPSCs for fundamental and translational discovery project. During this time, he has published in many prestigious scientific journals, including eLife, Journal of Cell Biology, Stem Cell Reports, EMBO, so on and so forth. He has previously received research support from Parkinson's Society of Canada and the Parkinson's Disease Foundation. He is currently funded through research grants from the Michael J. Fox Foundation, Parkinson's Society of Canada, Canadian Institute of Health Research, the US Department of Defense, and McGill Health Brains Healthy Lives Initiative. With this, I would give the space to Dr. Durkin. Please initiate or start your presentation.
Great. Thank you, Dr. Datta, for the kind introduction. Can you hear me okay?
Yes, we can hear you.
Perfect. And I'm just going to share my screen and then I'll get started.
Great.
Can you see my slides and everything okay?
Yes.
Great
Perfect.
stuff. So thank you and good morning, everybody. As you learned in the introduction, I'm actually based in Montreal, Canada. So it's the end of night here. So it's actually, you're a few hours ahead of me there.
Can you please make it full screen?
Yes, of course. Sorry about that. Do you see it now? Good.
Yes.
Yeah, so it's nice to actually be here. It looked like you had a great session of talks yesterday. So I'm sorry I couldn't be here, but I'm going to try, I guess, for today, kind of cover the work we do in our group focused on how we're applying patient-derived stem cells to studying the brain, brain disorders, and also kind of giving you an insight into some of the imaging we actually do in the lab to actually use these brain organoids to help understand brain disease in a way. So I guess the reason we got into all of this is actually over the last 20 years was we kind of would look at the field of oncology. On the cancer side, they've kind of over the last 2 decades really had an explosion in new therapies. And a lot of that's actually because we had an increased number of disease causing cells for each patient. So, patient with breast cancer, lung cancer, liver cancer, you always had more of the cells that had the disease. And it was quite easy to actually access the cells. So every patient had to get biopsied. You would then isolate them and grow them on a dish, and you could really deeply phenotype them and use these cells to understand what might be going on within the disease. And then you could then build and identify new therapies against it, from Keytruda to Opdivo to CAR T, to name just even a couple. We haven't really been as fortunate on the neurodegeneration side of it. So when we think about diseases like Alzheimer's, Parkinson's, you always lose the cells that you actually would like to study. Uh, and really, the cell types we would like to work with are pretty much lost, and they're within the brain itself. So it means they're kind of inaccessible. And so both with this kind of loss of the cells we would want to, coupled with how hard it is to get them, has really kind of made it difficult for us to work with human neurons on a dish itself. And so that's really been the crux of kind of both understanding and kind of developing new therapies for neuronal disorders. We want to work with human neurons on a dish, but we haven't been able to access them. And really, what we've been stuck to using has been these immortalized cancer cell lines like HeLa cells or other cells. And really, what we would like to work with are specific cell types from patients. So we'd like to work with human neurons or human liver cells. And to really do this, the idea of stem cells make this idea of creating human models possible, and many you heard yesterday and in other talks then today. Like, I don't think I really need to get too much into it, but just to kind of reiterate, what is a stem cell? It's a cell that can replicate itself, so it can divide and generate the same copy of itself, and it's pluripotent. So if you give it the right kind of developmental cues, you can then generate any given cell type that you may want, from a liver cell to a lung cell to even a heart cell. And so this kind of work all kind of really was pioneered by Dr. Yamanaka and his group back in 2006, where he kind of discovered the four programming factors, the Yamanaka factors, that would actually take a skin fibroblast from a mouse, and then it would reprogram it into what we now know as an induced pluripotent stem cell. And then a year later, obviously, through the work from Dr. Yamanaka and Dr. Thompson, then they're able to then generate human iPSCs from a human skin cell. And this then really opened up the field over the last kind of 13, 14 years, where now we have these style cells we can make from any individual. We can now make any cell type we want, either of an endodermal, mesodermal, or ectodermal lineage. And the Nobel Prize was then awarded to Dr. Yamanaka and Sir John Gurdon in 2012 for their work in iPSCs and cell programming. So really, that's where it all begins for us at the Neuro. So the Montreal Neurological Institute, it's an actual combined hospital and research center. We have patients with Parkinson's, ALS, MS, to name just a few of these disorders, that enter our door every day. And we're actually, through our open science framework, we're able to actually go to consent patients and then collect their blood. And so any patient that comes through the door, once they collect their blood, we can then access it in the group that we work with in the early drug discovery unit. And we've built up kind of processes over the years that from this blood, we're able to reprogram it into an induced pluripotent stem cell, or iPSC. Our groups now, we started in 2015. We're now 6 years later, about 40 plus people, kind of covering four main disease areas. iPSCs are at a core of everything we do. So we do both iPSC generation, phenotyping, CRISPR editing. We have an entire group dedicated to both discovery assays, screens. We do work with a lot of industry partners and also academic users. And more recently, we've really gotten into both organoids and tissue engineering, but really with a focus on brain organoids themselves. And then in the fourth group, we've had a kind of training and outreach. So both in terms of how we work with the cells, and then even the seminar series as well. But really, for a lot of the talk today, this is going to be the group I'm going to focus on, and kind of some new stories that have come out related to how we work with these brain organoids and what we're doing with them. And so we do a lot of work where we do generate 2D neurons. And more recently, we've really tried to couple it up so that if we were working with a particular cell type in a flat 2D dish, we would then have the neuronal organoid to go with it. And that kind of comes about advantages and challenges as well. And we work with three main disease areas: Parkinson's, ALS, and intellectual disabilities, and autism. And we try to have the two cell types that would be kind of the disease relevant cell types for studying the disease. So When it comes to Parkinson's, we primarily work with dopaminergic neurons. And then when we converted it to a 3D model, it would be a kind of midbrain organoid where it's like this 3D structure enriched within dopaminergic neurons. When it comes to the ALS side, we work with both motor neurons and astrocytes, and then we've also started to make our own kind of 3D co-cultures where we mix them together to form what we call these motospheres, or sometimes when we mix in the astrocytes to be called astromotospheres. And then when we're on the developmental side of it, we do cortical neurons and oligodendrocytes, and then the cerebral organoid then is matched in with it. But really for today's talk, I'm just going to focus on this side of it here and all the work we kind of do related on the PD side of things. And so, maybe for those that aren't super familiar with it, when it comes to Parkinson's disease, it's the second most common neurodegenerative disease, but worldwide and in Canada today has a prevalence of about 1.6 per 100 individuals, and it is a disease of aging. So, as people actually get older, so if you look in people over the age of 80, you do have an increased incidence as well, around 3.5 per 100. Symptoms of the disease include both tremor, slowness of movement or bradykinesia, muscle rigidity, gait, and you also have some kind of preclinical symptoms such as depression, anxiety, diminished sense of smell, even a disruptive sleep pattern that all kind of almost coincide or kind of manifest with the disease before you really get a kind of clinical diagnosis of PD itself. But there are really two kind of classical hallmarks of the disease. One is a loss of dopaminergic neurons. So when you have lost about maybe 50 to 70% of your dopaminergic neurons, then you start to get the disease. The kind of a lot of the motor symptoms start to manifest. And it's really motor, or it's really dopaminergic neurons that are actually located here within what is called the substantia nigra region within the midbrain of the brain. And so you can kind of see what this may look like or how it's reflected within the brain itself. You'll see kind of sections from a postmortem from an individual that was healthy and an individual that actually had Parkinson's. So you'll see this kind of black pigmentation here, which is neuromelanin granules. And this is like a byproduct of the dopaminergic neurons. When they secrete dopamine as a transmitter, it actually starts to form these granules and they can be stained quite readily as a kind of black pigment. When you actually start to lose the dopaminergic neurons themselves, you actually start to see this coloration actually starts to disappear. It actually diminishes in size, and then gradually, as the disease progresses, as neurons die off, the pigmentation even decreases more and more. And so there's actually a second hallmark as well, and that's kind of will relate then to the work I'm going to explain during the talk. And the other that classical hallmark is Lewy bodies. So Lewy bodies are actually they're an insoluble clump of protein surrounded by some membrane structures as well, but they're really enriched for alpha-synuclein. And synuclein is one of the first kind of genes linked and identified with Parkinson's, and both ubiquitin and other proteins then go into it. But then, as you start to see both the disease of patients develop, and also as you actually look in kind of histopathological sections from patients after they've passed away, you can really start to see these inclusions both within the dopaminergic neurons and also within other cell types within sections of the brain too. And so this is really where it all comes to, for us. Like we kind of dopaminergic neurons are like our bread and butter cells, like from patient cells or even healthy controls. When we work with the iPSCs, we follow these defined protocols. And you know, if you want to work with dopaminergic neurons within your lab, you're free to. We have the protocols online here and here, and you can then readily image these cells as well. Once you grow them, you can stain them for markers like a neuronal marker like TUJ1, and you can even stain it for dopaminergic markers like TH, which is tyrosine hydroxylase. And these can be grown anywhere. This is just up to 6 weeks, but you can grow them even longer, anywhere to 8 to 10 weeks, depending on kind of the assays and what you would like to do with it. And then you can easily see the kind of wide axonal network and also kind of the green cells here for dopaminergic neurons. And so we do a lot of work on the 2D side. I'm not really going to go too much into it, but we've really integrated both automation and high content imaging where we can both grow these iPSCs, seed the neurons in multi-well dishes from a 96 to a 3D 4-well format, and it just then gets applied into screening modalities. So where we have assays focused on kind of image-based ways, such as looking at synuclein uptake, even the like I guess mitochondrial morphology, lysosomal activity, and this then allows us to find lead disease that we can then bring to focus on is patient-derived brain organoids themselves. And this has really kind of led us to kind of build a 3D discovery pipeline that over kind of we started about 2 years ago that now is kind of taking molecules and targets and trying to be advanced into hopefully the next few years into therapies and then gradually into the patients themselves. So why do we want to work with 3Ds? That's a good question. Why add this third dimension? And the reason is this is what they would actually look like. This is actually just a whole man's what it would look like if you just fixed an organoid. And this is actually a section through an organoid. And you can see kind of these structures here where the neuronal rosettes emerge from. And then in yellow and green is actually dopaminergic neurons actually around the outside of a midbrain organoid. One of the reasons we actually like to work with this is it's very interconnected. So as you kind of grow and mature them, you have a kind of a lot of neurons spread across the different structures that can both interact with itself, but also interact with a lot of the support cells that are actually within the structures themselves. It's quite scalable, so you can actually make quite a lot of them, and you can then have quite like large amounts of human neuronal tissue. And that's kind of almost more reminiscent of what you would see within the brain that you can then really do deep phenotyping. And I'm going to kind of explain some of the uses you can do for that. They're quite easy to maintain, so that means you can really take them and grow them over multiple months. So when it comes to 2D neuronal plates, we can only really ever take them for maybe 6 to 8 weeks. When it comes to midbrain organoids, we've actually taken them up to 18 months. And all it really needs is once they're growing, you just have to do a weekly media change every Friday. So it's quite straightforward to do once you kind of have the team and the reagents that you would need to do it. And they're quite diverse in cell types, which then means you're actually getting tissue that's almost more similar to what you would see within the brain itself. Our interest is actually because we're working on looking at Parkinson's, we want something that resembles the substantia nigra as close to it as we can get. And because these are quite diverse in cell types, you actually have a lot of glial cells, astrocytes, and the neurons at the kind of the right percentages that you would actually see within your brain itself. And it's not just a midbrain organoid. It's actually one of the features of organoids is they're customizable. You can actually change the ingredients, you can change the factors, and you can direct it to make a different organoid. So you can have it more cerebral in nature, maybe forebrain, you can make a hippocampal organoid. And so this really then opens up the avenue so that if you think about different regions in the brain that you may want to study, you can then generate them and then actually grow them in a dish to then test therapeutics, which is really the ultimate goal is to both understand why a disease happens and then to see can we find something that can treat it. But I guess I always like to come back to this quote from George Box. Essentially, all models are wrong, but some are useful. And this is really where we're kind of at with brain organoids. There's actually, they're not the perfect model, but they do have a lot of advantages, but we'd also have limitations as well. We have to recognize it when we go in and work with them as well. It's a new field. It essentially only really came about in around 2013 when Dr. Lancaster, when she was working in the group of Jürgen Noblik, actually kind of was able to show from an iPSC line you could then generate these cerebral organoids that had a lot of the features of both the kind of cortical layering, a lot of the cortical and cerebral cells, and even could actually mimic and model microcephaly when you actually gave it a mutant line that had a mutation linked to microcephaly, you actually did see a diminished growth rate of the organoid and also kind of impaired layering and patterning within it. And so this really kind of led to a big hype that emerged probably since 2013, 2014 on brain organoids, where everyone kind of had this idea. Now, once we can do it, we can really start to clone brains on a dish, on demand, even that, from every person, we can get their brain tissue and then we can do all kinds of things. Like we can treat Alzheimer's, we can treat epilepsy, we can model Zika virus, for instance. We can even understand the development. There was even a recent paper from the Lancaster group that showed if you actually section through one of these organoids, you can actually have it connect to muscle, and then it can actually almost control the muscle contractions itself through stimulation. And then people have even taken it one step further. So this is actually kind of just highlighting some of the work from Dr. Alison Mottry's group at Stanford, where they actually grew a cerebral organ matured for over 6 months. And they showed actually, I think it was even up to 8 months actually, and they showed that as the organoid aged and matured, you really started to see an increase in firing. And this firing seemed to actually occur with almost like a pattern firing, similar to what you may see within a developing fetus. And so that really started to kind of lead to, I guess, this kind of concern of since we know we have this firing, should we work with it? And it also led to some kind of deeper studies into the organoids themselves that when we're growing, we're kind of thinking that they're almost this perfect tissue, but people in the field already kind of knew we had problems with it. We already had stress within the center, and that really just kind of got published last year when this paper came out showing that you had cell stress and cortical organoids. It wasn't really novel or kind of out of the blue. It actually was a very nice and elegant study. But really, what it showed was, and maybe it just brought kind of doubt for other people and maybe not working on it, was that there's imperfections in these mini brains or brain organoids. And this is true. If you work with a brain organoid, you have to recognize a lot of it relates to how an organoid interacts with its environment. It's always in constant shaking, and both how the media and the nutrients interact with it actually affect how it grows. And actually, in the center or the core of an organoid, it can sometimes be a little bit more stressed than what's actually outside and more the exterior region of it. And so the way I look at it is this idea of the cell stress isn't actually a bad thing. It just means that we're kind of very early in the whole process. So many of you may know this guy, Steve Jobs, and maybe some of you have also had an iPhone over the years. I know many of us now have smartphones in our pocket, but the kind of evolution of smartphones has always been iterative. You always had one or two kind of additions to it, either like a new camera, a new function, new memory, battery life. And gradually, what started here in the first version 13, 14 years ago is now completely unrecognizable from what you now have today. And that's kind of where I always see brain organoids, that we're kind of about 7 years into the process and we're only maybe on version 1 to 2. And we probably have many versions to go before we even get what we would actually think would be a human brain on a dish itself. And so, we're, let's say, on version 2. How would we even get to version 3 or beyond? And there's kind of several kind of factors or several kind of ways I think, or many of us think, we could do that. And one is both through bioreactors, where we enhance the growth and the kind of maturation of these organoids in a more uniform manner, through fluidics, so where you kind of actually bring in more engineering to enhance both the fluid flow of nutrients and air through the actual organoid itself. Better characterization. So I'm going to kind of talk a little bit about how characterization can be done about through imaging, but also kind of a single cell processing, and to actually take advantage of really the expertise on the tissue engineering and bioengineering side to making new devices. With all this kind of advent in 3D printing, we can now print whatever we kind of imagine. So we can actually print devices that can customize and grow organoids in manners that maybe we couldn't do 2, even 3 years ago. And also to really kind of think about how biomaterials can be applied to maybe make this tissue more like what you would see within a brain cell. And we're going to give a kind of example of a study we're doing to kind of move away from maybe the conventional hydrogels that we use, such as Matrigel, to maybe make materials that are more physiological to kind of the environment that would be in the brain itself. And gradually, the goal is really to get to what I would see as like almost an organoid toolkit, where you would work or want to work with organoids, you almost just go on a website, click a link, and there's kind of companies and groups out there that would essentially be making them. That could be just, they could ship them directly to you. And then you have your media, you grow it up, and then you're ready to do your experiments in a day or two without having to do all the hassle of having to learn it from scratch. And so I think that's going to probably be where some of the field will go over time. There's already companies that do this, like from Stemonics, where you can get these kind of neuronal spheres sent to you for screening. But I think this is going to advance more over the next decade. And so, really, kind of, these are three of the features I'm going to just kind of touch on about some of the work that we do and how to kind of improve organoids for what we want to then do. What is the biomaterial? So this is actually work we're doing with Dr. Chris Morais and his student Camille Castle de Camps, who are Chris is a professor at McGill like myself, and so he's actually a bioengineer. And so we kind of got together as a partnership, actually academically wise, 2 years ago, to actually kind of study the effect of different biomaterials. And in particular, one was alginate. So alginate's a very tunable hydrogel that you can actually mix it with matrigel, and then you can actually affect its stiffness. Because I guess what we were thinking is, when you press down on a brain organoid, it's actually very soft and squishy. But if for anyone that's ever touched a human brain, it's actually a lot tougher, it's a lot denser. And so we kind of wanted to see is could we more replicate that kind of stiffness and those properties of what you would see with a brain organoid. And that's kind of where Camille came in, where we actually took matrigel, where we would just grow it normally. And this just shows you the normal growth profile here. And then we started to add in alginate at different percentages. As you increase alginate at higher percentages, then you actually cause stiffer gels to form. And this is validated through both rheometry and also EM to kind of look at the pore structures. And when we actually had the stiffest hydrogel here, you'll actually notice that the organoid, the growth properties are actually much lower than what you would actually see normally, where it may actually be more uh, like a kind of normal organoid protocol. Camille actually did follow up this a little bit more in detail, where you would actually then show that both the size of the organoid would decrease, but also the number of these neuronal rosettes. And a neuronal rosette is really a kind of structure where these progenitors would then derive from, and they would actually be kind of where the neurons would actually develop from. But she noticed that as you actually started to affect the stiffness, you actually had less of development and less of these kind of rosettes that would form, which would almost imply that we were actually getting less development, which was then leading to less kind of growth size over time. And that's probably why the stiffness impacts development and then leads to a smaller organoid that then would form. And so this work is actually in the final stages of being written up, and we're probably going to be submitting it online over probably the next 2-3 weeks for those that are interested to see more of it. And the other area we've really kind of worked on in the lab is we started making brain organoids maybe about 3-4 years ago. It was actually a postdoc that was working in my group, Dr. Ngoyin V. Mohammed, along with actually Megar and Paula Lapine, who kind of joined the organoid team and really now lead it. That when we started working with brain organoids, we're only making a few hundred at a time, but we had many projects that had kind of needs for them. And so we needed to find a way to scale this up. And that actually led us to partner with a Montreal startup called Nuvio, where they had there we were and we kind of worked with them to make what we call these EB discs or embryoid body discs that would actually allow us to almost customize the generation of an organoid, where when we would dissociate iPSCs, we could then make these kind of 3D structures that would then be kind of placed in these honeycombs, and the honeycombs then eventually then gets deposited in the bioreactor. And this is actually a 3-month old, this is a bioreactor of 3-month old midbrain organoids that it's just one of them, but we can hold 9 of them at a time in an incubator. And we have about 5 or 6 incubators. So gradually, by kind of scaling it up, we have the capacity then to make close to anywhere from 14 to 15,000 organoids at any given time, spread across multiple experiments from multiple lines. And then by having this, that gives us some precedented material to then really do a lot of characterization into what's going on within the human brain. And that's really what we want these for, is like we want to build patient profiles on our organoids, and that comes from the patient iPSCs we have, that once we get these lines, each patient we can make their organoids. And then once we do it, we kind of like to ask 2 questions. One is, what do they look like? And so we've been really kind of building in a lot of advanced microscopy and even tissue uh, treatment techniques in the lab. One is like we do both histology, so paraffin embedding, and also cryosectioning. And so this actually just shows you kind of a cryosection through what a brain organoid would look like, where you have the kind of neurons and you even have the astrocytes all spread throughout the structure. You can even do whole mount staining of them, where you can just fix them and look at what the structure would look like just by eye. Or you can even go into more advanced or in-depth microscopy, where you could actually take these organoids and then you can actually do kind of 3D reconstructions both through confocal and light sheet microscopy. There is a question, so maybe what I'll come to the question actually maybe at the end, or I don't know. How do you like to do that? Actually, do you want to do the questions now, or how does the normal process?
No, at the end.
Okay, not a problem. So that would be kind of what do they look like, and then the other thing is what are they made of. So in a way, it's kind of like we think of it as almost like a cellular jigsaw. So I don't know if you're familiar with M&Ms or Skittles, where you have different colors. So I always think of it as like a brain organoid is almost like a bag of Skittles. We have all the colors under the rainbow, but if you squash it together, it all just kind of forms one homogeneous mess. But that gives you some information, it gives you some taste, but you lose a lot of the kind of information of what you already had. And that's kind of where we kind of come at it and we think about a brain organoid, that when you look at it, you kind of see it's complex, but you don't really understand the complexity until you break it into its individual components and then start putting it back together like a jigsaw. And by doing this, you can actually really start to see is what are the kind of the cell numbers, the actual cell varieties in it, and even understand is from a disease versus a healthy individual, is there changes? Do you see certain maybe genes that get turned on in a given cell type? So let's say if we have astrocytes, do they have more reactive profiles relative to what you might see within a control environment? And then this starts to allow us to make predictions and help to model the disease better within a dish by combining both the multi-omics with the imaging itself. To give you an example of what the imaging we do actually within the lab when we comes to 3D imaging, this is actually led by our imaging specialist, Dr. Maria Lacalle. So we actually, when we work with an organoid, we have to do specific treatments to work with them. So you always start with your kind of I guess, your white or opaque, uncleared organoid, and you then have to do a variety of treatments to then be able to do 3D reconstructions. And so there is kind of processes or methods out there for clarity, but the method we really use is called cubic, where it's just a variety of different ionic detergents to help remove the lipids. And then by the end of it, after about a week or two, you now have these kind of translucent or opaque 3D structures that you can almost see through. And once we have it like this, you can actually then stain them with different antibodies and really then get a sense of what an organoid may actually look like. Let's see, this is actually just to kind of show you how you can actually then image right through the organoid, where you can start to see in both red and green two different types of neurons. So I'll play it one more time. You can start to see in green the dopaminergic, and then actually, no, sorry, the red was the dopaminergic, and green was kind of just the regular neurons themselves. And then you can even then do a 3D reconstruction, where as you start to kind of take all the images and combine it, you can then rotate it and even visualize different features and start to see what the actual cells look like. So we've actually gone and taken it one step further. So we've actually done further clearing that's now gone onto a light sheet microscope. So that's actually in process. So we should hopefully have that data probably by the end of the month, which will then give us a lot more resolution of how both glial cells and neurons are actually kind of localized and adjacent to each other within one of these 3D structures. And so this is all well and good. So we can make these iPS-derived neuronal organoids. They grow. They've got neurons. They're active. So we have a whole kind of other data that we can show where you can actually put them on a multi-electrode array system, show neuronal firing, and you can do all of these things that kind of have features of the brain. But really, we want to ask, can they model the disease? And so our question really came because we're a specialist, or I guess one of our specialty diseases is Parkinson's disease. We then wanted to ask is, could we make a midbrain organoid or an MBO to actually model PD within a 3D model on a dish? And so this is really work that was led by Dr. Ngozi Muhammad that really started about 2, 3 years ago. On these are all just the kind of people involved with different parts of the project, from the flow cytometry, single cell profiling, to the histology, to even CRISPR editing and QCing of the cells themselves. And we kind of went in with question of, could we actually build a model that had characteristics of PD? So we knew from the human disease, you would always have these neuromelanin granules that would form. And so we wanted to see is, could we have a model where we could actually see these form? But the two main features we really wanted to see is, would we have the pathology that was associated with the disease? So I kind of showed you Lewy bodies kind of earlier, but Lewy bodies form from synuclein oligomers. Oligomers are almost a seed that they do form, and then they start to induce the formation of phosphorylated synuclein aggregates. And then gradually over time, as these form, you start to see a decrease in dopaminergic neurons, and then the disease starts to advance. When it comes to a rodent model, you can get some of these features, but you really have to almost give it like a molecular hammer. You have to give it like a preformed fibril, or you really have to almost give it a toxin to do it. It doesn't always happen as readily as we would like. So we just kind of wondered what would happen if we actually had a PD associated mutation within the model. Could this then lead to this happening? And so before we get into how we kind of built the model itself, this is actually the process of how we make these organoids. So we actually start with the patient iPSCs themselves. We grow them on just flat 2D dishes, and then we actually dissociate them, and then we actually induce them to form aggregates in kind of low attachment multi-well plates, or within the EB discs that we talked about earlier. Once these EBs form after about 5 to 7 days, they form just a kind of cell aggregate here. And once they do, you can actually then grow it, and then you can actually start to see its maturation. And so this is actually just shows you what it would look like after about day 15. So once you embed it in Matrigel, it actually, the Matrigel kind of gives it a support to then be actually placed within the kind of shaking bioreactor. And here you can actually see what it looks like at the beginning. It's only maybe 400 or 500 microns in size. And then after about 30, 40 days growing with constant shaking, it reaches its full size. It can't go anywhere beyond 4 millimeters in size. And now we've kind of shrunk it a little. So it's only about 2 to 2.5 millimeters, because we would notice that the bigger they are, the more necrosis and cell stress you had in the center. So that's kind of why we kind of shrunk it. So we'd still have the same cell number, but it was a bit more homogeneous and it was a bit less stressed. And for those that ever just wondered what they look like, these are just kind of what you would have as an example of a full-size midbrain organoid. And so this actually is all online in our kind of methods paper right here. You can actually then take these organoids, you can do cryosectioning through them and then stain for a variety of different dopaminergic markers. So we always OCT embed the organoids, and then we section anywhere from about 10 to 14 micron sections of the brain organoid. It's a bit different to tissue compared to what you may work with if you're doing mouse histology. But once it works quite well, then you can stain for markers like FOXA2, MAP2 is your neuronal marker, and you can then even see the dopaminergic markers kind of scattered throughout on the surface. And you can even then hone in on specific regions and really see the kind of the TH positive neurons actually within the organoids themselves. And you can even then take these organoids and associate them to single cell. And from here, then you can build a kind of jigsaw of it and then actually start to see profiles of it where you actually, from these organoids and the protocol we're following, we have maybe 20 to 25% of the cells actually in there are dopaminergic neuron, which is actually pretty close to what you would see with a substantia nigra. So there was a recent study that came out showing you actually had single cell sequencing, and it was maybe 20% of the cells were dopaminergic neurons. So we're getting pretty close to what you might have within the brain itself. But then there was a lot of other cell types there as well. So about astrocytes, oligodendrocytes, radial glia. The one cell type we don't have, which is something we are working on to see can we get it in, is microglia. And so that's kind of will be kind of something that's coming next, I guess, from a lot of groups. One of the areas we really wanted to confirm was to show could we actually you have dopaminergic neurons, but are they functional? So, i.e., if you give it dopamine, will it go through and actually do the entire dopamine metabolism pathway so that by going through dopamine, actually oxidize it and then eventually start to form these kind of vacuoles? So, when they form and enrich with iron, they kind of seem to form neuromelanin, which gives it a kind of a black pigmented color. And so this is kind of the test we do. When you normally have an organoid at 35 days, they're white and translucent. But if you actually give it dopamine for about 10 days, they actually turn brown in color. And so that's actually kind of showing that the metabolism of the neurons themselves are really active. They're actually able to metabolism dopamine. And you can even visualize it through histology. So you can actually parafilm embed an organoid. You can actually do sectioning on a microtome. And once you then do the sections, you can then stain for a dye called fontanemescin. So fontanemescin stain actually stains for kind of the melanin in a kind of different structures, and you can start to see these little kind of black dots throughout the structures, which are actually the neuromelanin itself. You can quantify it through different macros, and you can even visualize it close up. This would be what a neuromelanin granule would then look like here. And so we kind of knew we had this model now, the midbrain organoid, where we had neuromelanin could form, the dopaminergic neurons were active, they were even forming neuromelanin granules. So we then asked what would happen if we did something that was kind of related to what you would see within Parkinson's. So we actually went back to the first kind of known mutations found within Parkinson's in the late 90s, and that was a triplication event that led to increase in synuclein levels. And we actually were able to, through the Thilo Kunitz's group, find SNCA triplication patient-derived iPSCs that we could actually then use to model the disease itself. And so thankfully we were able to get 3 lines from him, actually a triplication line where we had 4 alleles. We were able to get an isogenic control where 2 alleles were actually knocked out. So then we would then expect it would have less synuclein expression. And then we also had a knockout line, which we've used in a lot of the validation experiments for the reagents. Um, Unsurprisingly, or maybe surprising, it depends on whichever way you come at it. If you'd had the synuclein triplication, so the Parkinson's line, and compared it to its isogenic control, it actually grew pretty normally. When you actually look at the growth profile over a 30-day period, you don't really see anything overtly different with it. We did kind of size profiling where you would just take them into wells and take pictures of them through the microscope, and then you would see that at about 60 days or even at 100 days, the organoids are actually pretty similar in size. There did seem after about 100 days, so 3 months growing, that you did start to see a significant decrease in size. And you can even visualize it here. And so we've actually kind of got some macros that we use with ImageJ that we can actually take pictures of these kind of like this and then quantify just the kind of overall size and calculate the diameter. We then went back to kind of the photoluminescent staining and even showed that in a kind of 50-day-old organoid, pretty much within the triplication versus the control, neuromelanin granules would pretty much form normally, and also the TH levels were kind of indicative of the dopaminergic neurons seem to be pretty comparable. You could actually do a comparison where you would actually look at both TH for the neurons of dopamine neurons and then MAP2 just as a neuronal stain, seem pretty comparable at just the early 50-day-old organoids. And so we wanted to ask then, okay, we know that these organoids are forming, and we know we have this mutation. So are we getting increased levels of synuclein? And thankfully, we actually were. So it all seemed to play out as we would have expected that when you have a triplication line, so you had the organoid and you looked at the transcript level RNA. So you just take an organoid and mash it up and then just look at the total RNA. The synuclein levels were elevated. It was decreased in the isogenic control and it was absent in a knockout. And this seemed to play out as well, consistent by Western blot. So we actually just blotted for a total alpha synuclein and we saw increased alpha synuclein in the triplication, a decrease in the isogenic, and no synuclein really in the knockout. And even further verification came at the histology. So they actually took the organoids, actually sectioned them and looked for total synuclein. It was actually highest in the actual triplication, and really the signal was absent when you then looked in a kind of knockout here. So you can see when you even zone in on particular regions, you can see these kind of red punctates for the synuclein in the neurons, and these were absent then really within a knockout ICC. So now we had this. So we then said, okay, well, we're not seeing huge effects at, let's say, 50 days. What would happen if we age these guys? And so this is actually what we did. We actually went from 50 days up to 100-day-old organoids. And when we did that, we actually found similar effects. We also, again, saw this persistent increase in synuclein levels. We did 2 different antibodies showing the test, and everything seemed to play out pretty similar to what we found before, where you would actually see the synuclein levels high in the triplication, both by blots and even by ICC, and you could then quantify about here and here. And then we were able to really zone in on certain regions, both within the organoid for the total synuclein, both within MAP2 staining and also the S100 beta staining. And so what we saw then was when we actually did the alpha synuclein, you actually saw quite high here, but we also noticed these kind of little kind of red dots or almost punctate that would seem to start to form within 100-day-old organoids, and they would start to kind of colocalize about MAP2 neurons and S100 beta glial cells. So we decided to kind of hone in on this and really look to see what might be going on. And so we kind of were able to take advantage of some of the stuff already in the field that we knew. We had reagents that could detect this phosphosynuclein. And phosphosynuclein is a marker that many use for actually detecting the pathology of Lewy bodies, so that when aggregates form, they're phosphorylated at synuclein S129. And so you could then detect it by Western blot, where you could see that within a triplication, with elevated synuclein, you also had elevated phosphorylated synuclein as well, which would indicate that you're also getting increased aggregates that would form. And indeed you do. You can actually see these aggregates that would form. The one thing that we have to point out, though, is that the aggregates are actually very dispersed. They're very sparse throughout the organoid themselves. They're not like spread throughout. You actually have to, they're in defined and discrete regions within 100-day-old organoids. And that's something that we're still curious as to why. Is there something about the specific regions where they form, or is there certain stress or something about it that might explain why they develop there and not in other regions? But you can really then start to zoom in, and then you can visualize and quantify the amount of phosphocyn relative to the total area. And you can even see it even if you do a much kind of closer up imaging on a confocal, where you can see these kind of red phosphosynuclein aggregate punctate co-localizing with both MAP2 and also within GFAP, which was another marker for the astrocytes. So as we aged to 100 days, we were able to detect phosphorylated aggregates in both neurons and glia, although kind of still at very low levels, maybe less than 1% of the total kind of area or the total cell number, but it was actually forming and it was present in both. I also have to point out, though, this isn't a cell type effect. So at the moment, every result was really in one triplication iPSC. So we asked, what about a different iPSC line? And so this is actually what we did. We went and found a different patient line that also had a triplication. So same mutation, but different patient, different genotype. And then we made the organoids again to 100 days. And this time we actually then did some kind of more powerful embedding and sectioning to see, could we see this pathology again form? And indeed, if you actually compared it to just a healthy control where synuclein levels are normal, you could actually visualize these kind of punctates throughout. And this just shows you kind of just some examples of what you would then expect to see in a kind of paraffin embedded section, and kind of similar to what you would see with a kind of cryo section. So kind of now we kind of knew we could do it by microscopy. We also then wanted to kind of go back more to a single cell approach to kind of maybe kind of gather some of the final data related to this. And that's actually where we took advantage of the flow cytometry infrastructure within the group, where we took the organoids, really dissociated them into their individual components using a kind of the specialized kind of dissociator we have in the lab. And then we actually, once we had the single cell suspension, we were able to kind of apply a battery of antibodies and then put it onto our Attune NXT flow cytometer to actually then build profiles of the organoids through the antibody staining themselves. So we started with 100-day organoids, and we were able to then detect maybe 0.43% of all the cells were positive for phosphorylated synuclein. And then really when we looked in the isogenic control, the signal was almost 10 to 20 fold less at 0.029%. And when we even looked at the knockout, which we used to gate it, there was no signal actually present for phosphorylated syn in the knockout organoids themselves. And then V wasn't really content with just finishing it there. She wanted to say as well, this is 100 days and, pathology is there, but could we actually get it even higher? So we actually went and matured a new batch of organoids a little bit longer, even to 170 days, and actually stained for both synuclein and phosphocin. And we noticed as well that, when we actually had in the triplication organoid, anywhere from about, I think it was 12 to 15% of the cells had actually were positive for high levels of synuclein. And when this is compared to the isogenic control, it was almost twofold increase. But then we then went back and looked at again the phosphorylated aggregates that would form it again. It was actually higher. But the one thing we did notice is that when we actually compared the levels here versus here, it was actually double. It was about 0.8% of the cells now were actually positive for phospho aggregates, whereas before it was half that. And again, within the triplication, while we're still getting a little bit more, it was still much lower than what you would see within the triplication. So as you had elevated synuclein, you were getting increased pathology. And to kind of really confirm that this is really specific to synuclein positive cells, we actually kind of cross-correlated it or overlaid the phosphosynuclein signal onto the synuclein background and saw that any phosphosyn signal was only present in synuclein positive cells, which is at least reassuring that it wasn't some sort of kind of false artifact or kind of uh, effects of the process itself. And so really so far we've shown kind of three of the main features. We can get the neuromelanin granules to form. We can get the pathology of PD to happen. We actually can also get synuclein oligomers. We have observed those too. We actually do synuclein proximity ligation assay where we can show that it forms. I didn't, just due to time-wise, I didn't include that data here today, but if you want to talk about it more, I'm happy to discuss during the question period. But we still had one kind of outstanding question, which was, do you see a decrease in dopaminergic neurons? So as pathology increases, do neurons or dopaminergic neurons decrease? And so we went back again to kind of the flow cytometry approach and kind of took different organoids at 100 days and showed that within the triplication line, you maybe only had between 10 to 11% of TH positive neurons that were actually forming. And you might ask, why is that a bit lower? I think sometimes it was actually due to the fact that when we compared it to the isogenic control, there might be some stress. Here, we were always able to get maybe anywhere between 15 to 18%. So it was actually a significant increase in TH positive neurons within our isogenic control, but a decrease in the triplication line. We also then did further follow-up experiments where we took the 170-day-old midbrain organoids and actually did kind of different markers like CD56 and CD24 to really pull out the neuronal population, and really found a significant decrease in neurons that would be present in the triplication organoids relative to what you would see within a control organoids. But kind of surprisingly, maybe to us, was we actually saw an increase, whereas the neurons were decreasing here, the number of glial cells was actually increasing based on the number of CD56 negative, CD24 negative cells. So it's something we're actually following now, and it's kind of where we're going with this, is to kind of understand more how neurons are being lost and how an increase in pathology may be contributing to all this effect from occurring. And so this seems to be what we're showing now, is we can at least have a kind of pathology is happening within these organoids, and it seems to be coinciding with a decrease in dopaminergic neurons, but it's really just a start. It really kind of is kind of scratching the surface, and there's a lot more work to come over the next decade for this. One is really to increase the maturation of these organoids, because at the moment, we've really only taken it to maybe, let's say, 170 days. About, what would happen if we took it to 250 days, 1 year, 18 months? What if we mimicked the entire COVID lockdown, for instance, and took it up to where we are now? Is there kind of effects in how the pathology develops and in terms of the disease itself, and actually to really understand how the other mutations happen? So we've really only looked at one mutation. We are now starting to, as we've scaled up for capacity, actually are looking at a lot of different other mutations, from synuclein to parkin to LRT2, that have all been shown to be involved in the development of PD. So to understand is there like a one size fit all model, or do each genes kind of have different effects in the organoids? And then that'll help us maybe better understand the different disease pathways that PD can follow. The other area we're really interested in is actually understanding the involvement of glia versus neurons. So I know we don't have microglia in it, and that's something we're working on, but there are other glial cells, both astrocytes and oligodendrocytes and radioglial cells, that are in the organoids, and we think they actually may actually be contributing quite a lot to how the disease is happening and how they interact with the neurons themselves. And so that's something we're following up on, but also then trying to understand is how increased inflammation may affect the pathology. So that's something we're trying to do, is if we, let's say, give it kind of a microglial cocktail, like the Ben Barres cocktail, where you essentially give it the cocktail a microglia would add to astrocytes to make it neuroreactive, what would happen? Would you then increase neuronal loss, or even kind of increase neuronal or synuclein pathology that would form? And I guess the last thing is really everything we've really looked at so far has been focused on just synuclein pathology, but cells actually have a lot of stuff going on. They have a lot of pathways, organelles, from lysosomes to mitochondria to lipids. And so this really is stuff we haven't even touched on yet. And so that's really kind of where we need to start going, is to understand how, as PD develops, like how our lysosomes may be affected on all these different cell types within an organoid. What about the mitochondria? What about even the lipid content? So there's a lot of questions that are still there and outstanding, and not just questions to ask, but also to improve the model itself, to really better model PD and to actually kind of come up with the next generation of therapeutics for the patient. And so this is really the team here. So this is actually a collective effort. So this is the group that I lead with Dr. Fawn, the director. So these are all the various people involved in a lot of the projects that we work on, especially Dr. Ngoy Naveem Mohammed, who really kind of has led and really built the organoid team to what it is, but also Meghna Mathur, who really kind of oversees the entire generation of maintenance of all the organoids itself. And then there's, where's Paula? She is here, who really has kind of led and kind of implemented the entire histology platform. And then also Maria Lacalle here, who kind of is our imaging specialist, is building kind of the 3D imaging setup for us now. And so I'll just kind of pause here. I'll thank everyone for your attention. These are, if you ever kind of want to talk more, have questions, that's my contact details. This is just some of the funding sources, out to CHR, CQDM, and all the others. And this is just to kind of end, on the kind of dopaminergic neurons and what you can go, like, these are neurons that you have within your kind of brain that you can now grow in addition what they would actually look like in cultures. So I'll thank you for your time, and I'm happy to take any questions. So thank you.
Thank you, Thomas. It was a wonderful talk and very enriching too, be it from the 2D to 3D, and you have taken through all the pathological details, that be it alpha-synuclein phosphorylation, accumulation, and even the detection of the melanin. So we have few questions in the chat box. We'll take few of them. I think little time will permit us. So
Yeah, so I can go through I have them in the chat box. So I saw the first question was from Pradnya. So how would one account for the systemic interactions or signals to mimic the disease? So PD is not confined to the midbrain. So you are correct, actually. If for anyone that works on, and I kind of didn't get into too many of the details here today, but it is true that PD kind of follows a spreading pathway. It actually goes through different circuits, and it actually even starts even earlier within the gut, can travel up to the brain, and then can go through circuit after circuit. So it's actually something we're starting to learn. A midbrain organoid is really the cell type that is lost. So where you can have Lewy bodies and other stuff that would form in other regions, it's really the substantia niger which is most affected. And so we kind of wanted to understand why the selective vulnerability is there, but we do recognize that there are other areas involved. And I think that's something we're starting to think about, how we can maybe, generate cortical organoids and even hippocampal organoids and start to kind of link them in maybe even a kind of interconnected assembloid or kind of even a structured fluidic setup. Even that's something we're trying to build as a kind of brain to gut access where you could start to see as if the disease developed in the gut, could it then spread into the kind of neuronal context? So it's a good question, and it's something we're all kind of cognizant to do for now. There was a question from Saham. Regarding the substantia nigra 2D model, would it be possible to 3D print a midbrain architecture or scaffold with collagen or other ECM gels and grow neuronal cells, etc.? I think it's certainly possible. Maybe people are thinking of doing it for now. It's kind of a little bit outside our wheelhouse of expertise. We're more reliant on the normal developmental process to let us then form it naturally. Whereas to do this kind of, I guess, artificially is something people can do where you could put the cells, but it's a bit trickier for doing neurons. I think there are one or two companies that have started to try do it, but it's very hard to print astrocytes and neurons and all the stuff in a kind of the arrangement that you want, because neurons are actually very sensitive cells, but also to kind of have the axons grow throughout and start to interconnect is not something that's quite easy to do with this. But if you have ways to do it or are going to do it, I'd certainly love to see if it can work in time. Um, from Swati, do microglia develop innately within your organoids? So no, they actually don't. So microglia are mesodermal in nature, and we're primarily working with, I think, ectodermal tissue. So there has been a group that have tried to kind of make a modified version of the protocol. We've tried it as well, but it didn't really work very well. So we're actually trying to now more grow the midbrain organoids as they are and grow the microglia separate and then try to kind of mix them in together. So that's something we're working on actively, but we haven't fully got them integrated just yet. It's still probably too early to say if that'll work or not. And so do we kind of think they recapitulate at least part of the pathogenic inflammatory profile? So I do think they do, and that's because they have a lot of other glial cells present. Like we don't have microglia, but they do have astrocytes, they do have oligodendrocytes, radioglia, and some other glial cells as well. So I think there are kind of a lot of cells that can get the inflammation that maybe we don't see within PD. We do have a project, actually I have one student working on it where we're giving the microglia a cocktail that'll turn on astrocytes to then induce inflammation. So that's kind of where we're going with that, and we'll see if something comes from that. And that, so we'll hopefully know if you talk to me again in 12 months' time, I might have a better sense of how that goes. And then other questions. Saham, do you see neuromelanin pigmentation in a 2D dopaminergic culture? Not really, actually. So I think it's because of a maturity issue. So we could probably give it dopamine in mature neurons at 8 weeks, and then we might see a little bit. But when we just grow them normally by themselves, no. So we don't actually see neuromelanin form until after about 6 months in a brain organoid. So if you age it to about 6, 7 months, they actually turn brown naturally. When we do it at 35 days, we have to actually give it dopamine by itself to switch everything on. But when it comes to the cultures, I think it's just that 2D cultures just can't live up to 6 months, or at least they don't in our hands using conventional methods. But I think some people are doing it, but it's just not as easy to really do within a 2D culture. And then the last question from Pradnya, does the organoid mimic the neuron glia ratio? Flow sodomy does not clear. Yeah, there was a lot of other stuff. We have a lot of other data on the flow. When you say mimic the neuron glia ratio, I guess you mean what you would see within the cell type itself. So you do see it from single cell stuff. One of the limitations when doing flow is actually some of the antibodies and how well they work. you kind of see more of the ratios consistent when you do single cell multi-omics. And so that's something, we have a lot of other supplemental data that'll be going in the paper when V puts it out soon. So you can kind of see a lot of it there. And if you have questions or comments, we'd love to know more about it. The flow is something new that we did that we thought we'll try it and see how it goes. And so it's been quite useful, but there are still limitations, and a lot of that's really due to the reagents that you use for staining cells.
Thank you, Thomas. I have one, it was a great talk, and I have one question. So how do you go about for determining or detecting the electrophysiological studies on these organoids?
So we actually have a multi-electrode array system in our group. So we actually we work with the Axion Maestro system, and so we work with they send you kind of 24 well plates, and so we just kind of, once the organoids are ready to work with, we actually just place them down onto the MEA, and then we just record. So we have a kind of there's a protocol we follow. So it's actually done more with Ted Fahn's group. He is a student, Ghislaine, that's been doing them. And so as we age the organoids, we've done it up to 6 months now, you start to see an increase in firing and more kind of patterning with the midbrain organoid. We are kind of talking to some of the other companies, so Maxwell, Tree Brain, and others, about they have new systems and new models that at the moment it's this kind of flat probe system where you just place it and the neurons that are on the surface will access it. There has been companies now, and there's been papers talking about these kind of almost 3D probes that go directly and insert into the tissue itself. And so they've kind of approached us. We're looking at it, and we'll see maybe we may integrate it or not. I think one area I'd love to do is more like nano chips, where I could actually just make the organoids, but have the chips go directly into the organoid, so they would be touching the neurons at different regions, and then we could actually get real-time measurements of it. That's still my pipe dream. We haven't got there just yet, but I think there's been one group that have done these kind of what they call cyborg organoids, and so that'll be maybe something in the next few years we try to do, or maybe others kind of figure out how to do it, and we can work with them to do it ourselves. So yeah.
Thank you. We do use the MEA over here. So that was great having you here and learning so many things from you, and getting to know that in iPSCs and Parkinson's disease, which extent and where the neuropathology is getting mimicked in a petri dish. Thanks a lot again for joining in. I know the hours are not, it's quite late over there at your place, but nonetheless, we appreciate your participation.
Not a problem. I'm happy to be here and happy to present our work. And don't be afraid if anyone wants to reach out, and you can reach me. I think Dr. Datta has my email address, or if you have questions or suggestions or ideas, happy to talk more. So it was nice to meet you all today.
Thank you.
Thank you.
So from induced pluripotent stem cells, now we will shift to another stem cell-like cells, which are the cancer cells. So coming to that with our next speaker, we'll be telling more in details. So Dr. Sudha Warrior is an additional professor at Manipal Institute of Regenerative Medicine, Manipal University, Bangalore, a stem cell biologist working research in regenerative medicine from 2006 in India, US, and Australia post-PhD. She's also an adjunct associate professor at Curtin University Medical School, Western Australia. Her key areas of interest includes regenerative strategies using mesenchymal and pluripotent stem cells for cardiovascular and neurodegenerative disorders, and drug development targeting cancer stem cell with a focus on Wnt pathway. She's actively involved in mentoring students for research projects for PhD, MTech, MPhil, and master's program in projects related to drug discovery, neurodegeneration, epigenetics of cardiac and endothelial regeneration from stem cells. She has been one of the pioneers for the design of disease in a dish models of complex diseases like Alzheimer's, Parkinson's, and atherosclerosis. Having several patents and publications in the field, she has commercialized her research through her biotech company based in Bangalore, Cure Stem Cell Solutions Private Limited. She's also a recipient of commercialization grants from BIRAC, DBT other than academic grants from ICMR and DBT Stem Cell Task Force. She has been awarded the Certificate of Excellence in Healthcare Leadership from Curtin University Medical School, Western Australia, She has active collaboration with Curtin University, Australia, and National University of Singapore, and collaborate with scientists from Malaysia in grant proposals. Her commercial collaborators are spread across Europe, Singapore, Malaysia, and Australia. She's also a recipient of Indian Initiative Grant from Curtin University. Dr. Sudha obtained a PhD in 2002 from Madras, University of Madras in reproductive biology. She underwent postdoc in Duke University, North Carolina, in Medical College of Ohio in cancer biology. So with that, I would keep this open for Dr. Sudha to start out with her presentation. Please go ahead, Sudha.
Thank you, Dr. Indrani, for your kind introduction, and thank you also for inviting me to this exciting conference. As you rightly said, I will be talking about a different kind of stem cells which are not involved in regeneration. So I'll just share my screen now.
Yes. Yes, it's visible. You can make it visible.
Okay.
Great.
Okay. So the title of my talk is emerging approaches for targeting cancer stem cells via the Wnt signaling pathway. So here I will be touching upon what are cancer stem cells, what gives them the stemness potency, and what is the terminology, what are the regulatory factors of cancer stem cells, and what is our approach in targeting this elusive kind of stem cells, which is within the tumor. So cancer stem cells hypothesis itself deals with the notion that there is a resident population within all tumors, which is like a drug refractory, which is not, which is like untouched by the external environment, and which has a special niche area which helps in its sustenance. So the stem part of the cancer stem cells comes from its phenotype of being resistant to drugs. And it's also prone to active self-renewal on the one hand, and on the other hand, it remains quiescent. Unless it is provoked, it does not come out of its niche and does not actively proliferate. So when you look at the cancer stem cell model, you have two kinds of strategies. That is, actually, the existing strategy is that is, if I mean, like, if you are targeting the tumor bulk, then there will be retraction of the tumor bulk, which is the non-cancer stem cells, and there is a tumor shrinkage initially. But since the cancer stem cells are remaining in that population, there will be tumor relapse, because the more the cells in the cancer, I mean, more the number of cancer stem cells which are remaining within the treated tumors, more the possibility for the tumor to relapse. So the top panel shows the conventional chemo and radiotherapy. Whereas if you were to target the cancer stem cell specifically by addressing its unique stemness features, then you have the drugs, that is the chemotherapeutic drugs, being being able to enter the cancer stem cells, so that which is part of chemosensitization, and that could result in shrinkage of the cancer stem cells itself, along with the tumor bulk. So if you have a dual therapy, that is the tumor bulk being addressed by the conventional chemotherapeutic, and you have the cancer stem cells being targeted by CSC specific drugs, then that would eventually result in tumor resolution, and there will not be relapse. So here, what would be the parameters that can be addressed for specifically targeting cancer stem cells is mainly the signaling pathways that are unique to cancer stem cells. So basically, that could result in decrease of EMT, and definitely a reduction of angiogenesis and vascularization, and also decrease in the drug transporters. So, what exactly is the origin of cancer stem cells? There are different theories which propose that from where the cancer stem cells are derived. One is the terminally differentiated cells can de-differentiate itself and form the cancer stem cells, or the resident stem cells within each and every tissue which is present for repair undergoes dysregulated self renewal and could generate the cancer stem cells. Or the transit amplifying cells, so the progenitor cells, which is in between the completely differentiated, terminally differentiated cells, can undergo de-differentiation and result in the cancer stem cell population. Or even there can be trans differentiation from the circulating bone marrow cells or bone marrow stem cells that would result in the formation of the unique population of cancer stem cells. So here we are concentrating on dysregulated self-renewal, because that would be a perfect drug target to address to, rather than the other parameters which are mentioned here. So this is a variable by which we can target these specific cancer stem cells. So for the CSCs to sustain itself within the tumor niche, tumor itself, there is a niche area. As I told you, cancer stem cells are not rampant within the tumors, and that exactly is the reason why they cannot be easily targeted. But saying that there is a small niche within the tumor which sustains the cancer stem cells, and that niche area, if that can be actually busted, then that is also another therapeutic strategy to address cancer stem cells. So here we are looking at a niche area which has a high level of self-renewal pathways, such as the Wnt and the Notch, and also that could result in the key features of cancer stem cells, key features which define cancer stem cells, that is the self-renewal, plasticity, dormancy, metastasis, and therapeutic resistance. So when one needs to address cancer stem cells, then all these factors has to be looked into. There has to be a separation of all these factors, which would result in the destruction of cancer stem cells. And one such pathway which addresses all these key phenotypes of cancer stem cells is the Wnt beta-catenin pathway, which is what I will be talking about in this talk. What are the emerging pharmacological strategies for CSCs which include self-renewal pathways? So mainly these pathways can be inhibited by specific antagonists, and also which results in chemoresistant reversion, basically. So if there is a chemo sensitization, first and foremost, if there is chemosensitization, that would result in uptake of the normally used chemotherapeutic by the cancer stem cells. We are looking at specific antagonists for the Wnt pathway, which as I told you, suppresses the key features of cancer stem cells. So what is the Wnt beta-catenin pathway, which is a very well known pathway, which is activated in neurogenesis, and which has an inhibition in cardiogenesis, and different organogenesis pathways follow different protocols for Wnt activation or inactivation. So incidentally, the Wnt pathway is mainly upregulated in neurogenesis, and here when the Wnt is on, then you have the extracellular ligand, the Wnt itself, which binds to the frizzle receptor, and activates beta catenin intracellular signaling, which binds to the nuclear receptors, that is the nuclear factors, in fact, TCF, LAF, and initiates the target genes. And during its suppressed normal off conditions, then you have the phosphorylation of beta catenin, which eventually results in, which is a default state in which the TCF-LEF promoter region is attached to or is bound to Groucho, and there is suppression of the Wnt target genes. So here you can see the key player in the activation of beta catenin is GSK3 beta, which is inactivated in the on state, and that would result in dissolution of the destruction complex, which is then attached to the frizzle receptors. And that releases the beta catenin and results in internalization or nuclear localization of beta catenin and activation of the Wnt downstream targets. So our strategy is to target the self-renewal mechanism of CSCs via the Wnt beta catenin pathway using an antagonist, the secreted frizzle receptor related protein 4, which antagonizes the Wnt ligand itself, which I'm working in collaboration with his co-discoverer, Professor Dharmarajan from Curtin University. Presently, he is in Chennai. So this protein was originally reported in as early as 1997 as an apoptosis-associated gene. And it was discovered through a differential clone display. And incidentally, it was found to be a protein which is involved in most of the pro-apoptotic tissues, such as the reproductive tissues undergoing apoptosis. And in cancer cells, it was found to be not only pro-apoptotic, but also anti-angiogenic. And specifically in cancer stem cells, we have found that it increases chemosensitivity. So normally it works well as a combinatorial therapy along with other chemotherapeutics. So the protein itself is a 40 kilodalton protein, and it contains specifically two domains, active domains. The CRD domain, which is the amino acid terminal domain, which is about 120 amino acids, and the natrium-like domain, which is towards the carboxylic end. And incidentally, both of which have individually itself have a pro-apoptotic property. So Wnt antagonist, how does it act? It can bind to the Wnt as shown here, and suppress the downstream targets of the Wnt genes. Or it can also bind to the frizzle receptor itself, and thereby blocking activation by Wnt. Incidentally, the SFRP4 I just mentioned, it has two domains, one of which is the CRD domain. CRD domain has homology to the frizzled receptor itself, as shown here. And there could be a dimerization of the CRD with the frizzle receptor, thereby blocking the activation by Wnt. So both have an effect on suppressing the downstream signals of Wnt. So we looked on the SFRP4 and its activation first in head and neck cancers. So we found that cancer stem cells from head and neck cancers are chemosensitized by SFRP4 by inducing apoptosis, importantly decreasing stemness, which is why we isolated the cancer stem cells itself, and decreasing drug resistance, and also reverting the epithelial to mesenchymal transition. So in this study, we found that the SFRP4 inhibited the growth of CSCs from the two lines that we studied, that is the HEP2 and KB. We used the chemotherapeutic that is commonly used for head and neck cancers, namely cisplatin. So our take home in this report was that SFRP4 itself chemosensitizes CSCs to the oncotherapeutic cisplatin and decreases the ABC marker expression, which is upregulated, typically upregulated in all the CSCs. So that results in drug efflux, and the SFRP4 has the effect of in fact suppressing ABC expression itself via the Wnt pathway, because one of the target genes of Wnt are ABCG2 and ABCG4. So it also decreases it increases the epithelial-like status and reverses EMT. So with that finding, we looked into other solid tumors such as glioma, and we worked extensively on glioma cell lines, and we found that similar to the head and neck cancers, SFRP4 indeed chemosensitizes the CSCs from these gliomas to temozolomide. And it specifically inhibited the Wnt beta-catenin pathway, but surprisingly, it enhanced the Wnt calcium pathway. So it is now known that SFRP4 is a antagonist of the beta-catenin arm of the Wnt pathway, whereas it is a promoter of the enhancer of the calcium pathway, which also induces apoptosis downstream. And the combination therapy of SFRP4 and temozolomide significantly reduced tumor burden in mouse. So this was an interesting finding. And from these findings, we have now looked into most solid tumors, such as breast cancer, ovarian cancer, and others. And we found that across most of the solid tumors, the SFRP4 indeed acts as a chemosensitizer to the existing oncotherapeutic, and also reduces the burden of the oncotherapeutic to help in the reduction of the tumor size. So it helps in reducing the toxicity, and also helps in inducing. Also helps in basically suppressing the CSC population within the tumor bank. Now, we were interested in looking at some of the natural compounds. This I have done in collaboration with with the collaborators from NUS Singapore. Dr. Gautam actually and Dr. Allen, who are my collaborators, wanted to test the effect of a natural compound, that is diosgenin, on breast cancer stem cells. And we found that, in fact, diosgenin itself, which is derived from, which is the saponin, steroidal saponin, inhibits the CSC properties of breast CSCs, and specifically inhibits the Wnt TCF-LEF pathway. And the way it acts on the CSC is by, in fact, upregulating SFRP4 and also the other Wnt antagonist, that is DKK. So now when we compare the antagonism of the well-known antagonist, that is Dikoff, versus SFRP4, we find that Dikoff is only can act only through the beta-catenin pathway and not via the other pathways, and therefore it is rather limited in its ability for suppression. And more importantly, in the other studies which I'll just be talking about, we found that the domains of SFRP4 is equally effective as the whole protein in inducing apoptosis. So next we were interested in looking at what exactly could be the role of SFRP4 besides its obvious extracellular binding property to the Wnt ligand itself and the Wnt receptor. Because in some of our earlier studies, we observed that that SFRP4 is found, in fact, localizes within the nucleus. And so we found in this study, which we reported in cancers, that SFRP4 in fact has a DNA binding ability and activates P53 via upregulation of the pro-apoptotic MER, that is MER 8855P. And by this, so therefore, there was another link of SFRP4 to the apoptotic machinery, that is directly via the P53 pathway. And SFRP4 knockdown induces, in fact, pluripotency gene expression. So that was also that was also the link between the CSE phenotype and SFRP, because cancer stem cells itself are defined by its upregulation of most of the pluripotent factors such as OCT4, NAN, SOX2, etc. We found that in fact SFRP knockdown does reactivate pluripotency gene expression, and also SFRP knockdown upregulated the Wnt canonical pathway, whereas downregulated the Wnt non-canonical signaling. So, with that in place, we also looked at another natural compound which we've reported recently, that is ursolic acid, in collaboration with NUS. We found that this natural compound, which has no off-target toxicity, was in fact induced the inhibition of breast CSCs, and incidentally, that is activation of SFRP4 was also seen to be another effect of treatment with UA. So SFRP4 could be initiated by UA to function as suppressing the Wnt activity specifically, and thereby disrupt the target genes involved in drug resistance and self-renewal. And another mode of action of UA observed in the study is that UA targets, in fact, the another MER, which is pro-oncogenic, that is MER 499A5P, which is upregulated in several cancers, including breast CSCs. And Wnt pathway, in fact, inhibits the breast cancer stemness. So this is so we found that ursolic acid also acts via activation of SFRP4 and also via suppression of another pro-oncogenic factor, that is MER 499A. And now, like in the to look at commercial potential, or to look at how we can actually convert these SFRP protein into a commercial drug, we were supported by some funds from Commercialization Australian Bureau, by which we had to study the two domains of SFRP, and if these domains can be actually if we can resolve these domains into the smallest size amino acid length possible, smallest peptide possible, so that which peptide is most effective, what's the smallest size of peptide that is most effective in causing apoptosis equivalent to the whole protein. So here we isolated, we in fact designed some peptides, that is from the CRD region as well as the NLD region, the netrine-like domain. And we generated two peptides, that is SE301 and SE401 of 15 amino acid and 20 amino acid length. And we found that the SE401 in fact is pro-apoptotic, whereas SE301 also has an additional anti-angiogenic ability. So therefore, that would result in it being one of the designer drugs, because it can suppress the vascularization of the tumor on one hand, and also alongside suppress the proliferation process. So we found that in fact these peptides, when when these peptides were added to, in fact, breast cancer CSCs from MDAMB as well as MCF7, we found that there was complete disruption of the spheres, which is one of the characteristic properties of CSEs, to form CSE enriched spheres. So the sphere disruption was clearly observed with both the peptides, that is SC301 as well as SC401, in these cell lines. So here, when we subjected to the oncotherapeutic, also, that is doxorubicin, the disruption was fairly complete, which showed the ability of the small peptides also to act as effectively as the bigger domain or the whole protein. So another angle that we wanted to look at, that is with these peptides, that is SC301 and SC401, was in ovarian cancer stem cells. So here we wanted to see one of the defining feature of ovarian CSE is its active upregulation of CD24. So we wanted to look at whether CD24 was specifically downregulated by these peptides. And we found that indeed it was. That is, especially the netrine-like domain had a very active suppressing effect on CD24 in comparison to the 301. And in our subsequent studies also, we found that SC401, which is a 20 amino acid peptide, which is derived from the NLD region of the whole protein, was more effective than the other peptide, that is the 301. In addition to that, in ovarian CSEs, we also found that these SFRP peptides can lead to the downregulation of EMT and upregulation of the epithelial markers, such as ECAD, and therefore it also has a general the general suppression of the cancer stemness properties. And besides that, we found that in ovarian context, these peptides on its own, without the oncotherapeutic itself, was able to effectively disrupt the spheres, and also reduces its ability to migrate. So these are all key properties which would help in suppression of the CSE population. So our aim is to actually even avoid the oncotherapeutic itself. I mean, what is the lowest concentration of? We would like to titrate in such a way that we can get the lowest concentration of the oncotherapeutic, by which there is a suppression and a suppression with the help of the SFRP peptides. So now, with that, like with those studies of the SFRP whole protein and the SFRP domains and the SFRP peptides, in addition to the natural compounds that we used, we found a common line that that there is upregulation of the Wnt antagonist SFRP4 when the natural compounds are used, and SFRP4 by itself is a potent CSC targeting drug. So in those lines, we would like to, we are looking at developing small molecules from the short peptides that we have studied. But in addition to that, we were also interested in another pro-oncogenic factor, that is the dead box helicase, in collaboration with Dr. Allen from NUS. So he works extensively on DDX20. In fact, is one of the co-discoverer of this protein. And it was found that that DDX20, which is basically a RNA helicase, which supports the secondary structure formation of the RNA, is a regulator of the Wnt beta-catenin pathway. So what is DDX20 and how is it linked to the Wnt antagonist angle of study? So DDX20 is also called Gemin3, and is implicated in a number of cellular processes, including neurogenesis. It is a transcription repressor. It is basically a pro-oncogenic factor and drives the metastatic potential in breast cancers. Typically, in triple negative breast cancer, it is overexpressed. And the key feature of TNBC is that beta catenin signaling is pronounced. So this is a good model. We thought this is a good model to study the Wnt antagonism because that is the most active. Breast cancer stem cells obtained from TNBC are most active with most actively expressing the Wnt beta catenin factors. So DDX is an essential regulator of Wnt beta catenin signaling, as I just mentioned. And here we can see that that is silencing of DDX, siRNA of DDX resulted in decrease of beta catenin, and there is when there is silencing of DDX, active beta catenin is suppressed most effectively, which is brought back into action by the addition of Wnt3. So here we also show that DDX specifically depletes the cell cycle progression as seen in all these analyses. Basically, suppression of or siRNA of DDX results in decrease of the pro-oncogenic c-MYC, and we can see that cyclin D1 is also suppressed in both the cell lines studied. And there is definitely a halt in cell cycle progression and accumulation in the sub G0. The DDX itself is works on the Wnt beta catenin positive feedback loop, as shown in these graphs. That is, addition of the ligand Wnt3A will result in upregulation of overexpression of DDX. And inhibitor of Wnt3A results in specific suppression of DDX20. And also TCF4, which is the nuclear transcription factor, was also suppressed on siRNA of DDX20. So here DDX itself would in fact is a activator of the Wnt beta catenin signaling, and therefore it is highly expressed in breast CSCs. And here when you when there is knockdown of DDX, that has resulted in depletion of the key marker of breast CSCs, that is CD44. There is complete abrogation of expression of CD44 as seen here. And most of the pluripotent factors are also suppressed upon siRNA treatment of these breast CSCs. So this has led us to what is the role of what could be the link between DDX and SFRP4. So targeting DDX with SFRP4 is something which we were looking into, and we found that that is DDX itself would result in, if you overexpress either the SFRP whole protein or the domains, that results in a decrease in expression of DDX20. So here in this panel, you can see the different forms of SFRP, the whole protein, the NLD, the CRD, and the ligand itself. So Wnt ligand results, as shown in the previous graph, results in overexpression of DDX, and all these forms of SFRP has resulted in suppression of the DDX protein, thereby showing that there is a direct link between SFRP based Wnt antagonism and DDX expression. So the docking studies which was done in Curtin University also showed that that is Wnt3A, in fact, interacts with the SFRP-CRD domain. And that the physical interaction has been demonstrated here by docking studies. And here we can see that addition of SFRP4 results in decrease in DDX20, and addition of Wnt, in fact, as shown in the previous slide, results in activation of DDX20, showing that the feedback loop between Wnt beta-catenin pathway and DDX is a positive feedback loop, is operational, and therefore targeting the DDX positive population in triple negative breast cancer by SFRP4 and its domains and probably the peptides would be an effective strategy to address these cancer stem cells. And here we can see that depletion of DDX20 results in chemosensitization to dogs. With siRNA and DOX treatment, there is a complete reduction of cell viability, which is also reflected in the sphere formation assay, with only DDX20 knockdown and DDX20 knockdown plus doxorubicin. So the next line of research that is in fact funded now is to generate small molecules of the peptide regions, that is the NLD and CRD. So we have obtained a 5 amino acid peptide from the 20 amino acid which I was talking about. We have obtained the smallest version of the peptide, which is 5 amino acid, which is equally effective both from the CRD and the NLD domains in inducing apoptosis and in arresting angiogenesis. So the next plan is to design small molecules based on this interaction or based on this peptides. So to conclude in this study, targeting of CACs of gliomas using the SFRP4 is an effective CAC inhibitor. And there is decreased self-renewal and all the stemness properties such as migration and invasion. A surprising finding was SFRP is a DNA binding protein. And that work is actually still underway. So we don't know what exactly it does by binding to DNA. Is it linked to the TCF-LEF? Is one question, but we don't have the answers yet. And SFRP4 could also have novel functions, that is in upregulating the apoptotic MERV885, which in fact directs P53 activation, which is a well-reported link. And SFRP in fact could have a direct role in arresting the pluripotency factors. The pro-oncogenic MERV, which we observed in the breast CSC studies using ursolic acid, we found that that is the UA, natural compound UA, in fact targets MERV499 itself, and thereby results in abrogation of the CSC presence. So depletion of DDX20 reduces stemness of CSCs is the other link that we found out. And we suggest that a possible Wnt3A DDX positive feedback loop is possibly mediated by the TCF transcription. And Wnt antagonist SFRP4 and its associated domain, in fact, reduces DDX20 expression. So here we have talked about two oncogenic factors, that is MERV499 which is activated by which is activated in breast CSCs, and DDX20 which is specifically activated in triple negative breast cancers, both of which can be suppressed by the induction of SFRP4. And SFRP4 is suggested to block Wnt3A induced DDX20 expression. So with that, I would like to thank my funding agencies. That is mainly the peptide work was supported by Curtin Commercialization Advisory Bureau, and the funding is still on. The work was on DDX was done in NUS in collaboration with Dr. Allen and Dr. Gautam. And a lot of work on the small molecules has been done in Curtin University, and have also been supported by DBT and ICMR in these in the angiogenic studies. This work was done extensively, especially the pluripotency factor. I mean, like the ability of SFRP to bind DNA, and the other work in that paper was done by my PhD, a past PhD student, Bhuvna, and it's been continued by Saurabh and Mohan here. And the other collaborators are the other people who worked in this study are Sebastian and Abhijit, who are doing the postdocs in UK and US. And the work has I've been introduced to the Wnt antagonism angle of CSC suppression by Professor Dharma from Curtin, and also been actively supported by Frank, who's helped me with the manuscripts. And I would like to thank Nimhans and Dr. Indrani for an opportunity to present this work. I'd be happy to take questions.
Thank you, Sudha, for such an extensive work sharing with us with a lot of patience. And not only that, it also gives an opportunity to understand that with in vitro cancer stem cell as a model in the Petri dish, so much in detailed classification of compounds to its validity to its functional relevance can be unraveled. So thanks a lot for sharing your data with us. If there are any questions, you can put it in the chat box. Okay, so I have one question. Coming to your 5 compounds which you all are looking into. So ultimately, are you all beyond this cancer stem cell model? You all are next going into in vivo work, or how would you see that it is reaching to the tumor region and it is capable of bringing about the change there?
Yes, yes. I'm happy that you asked this question because we have already worked on this in animal models in the breast scenario as well as in gliomas. So the next thing is that, as I told you, like, the reach is fairly okay. Like there is actually tumor regression and anti-androgenic ability is seen in ovarian cancer, breast, as well as in gliomas. However, when you look at especially gliomas, this peptide is not going to really effectively reach the brain. So, and also there is, I mean, to increase its uptake, we are looking at actually generation of, on the one hand, encapsulating it. Those studies are underway with some novel kind of molecules. In fact, calirexin. We are constructing these peptides with encapsulated with calirexin and looking into its injection in animals. And on the other hand, we are in fact generating small molecules itself so that they can be directly put into preclinical trials, and those studies are underway. So we expect these small molecules to have a really targeted effect on CSEs. And our dream, in fact, is if it can, what we've seen in the Petri dish, if it can address CSEs, destroy CSEs with the minimal amount of oncotherapeutics, so that there is minimal off-target effects.
Yes, true. Best wishes for that. And with that, we will go for the next presentation, which...
Thank you, Indrani.
Thank you, Sudha, a lot for taking out time and joining our symposium. And you are most welcome to be there throughout for today's session. And yes, we will move on to the next presentation. Thank you. So good morning again, everyone. I think some of you all had already attended my talk yesterday. So I had covered on the little bit of work which my PhD student had done on in vivo imaging and how optical dyes and tracers can be used for understanding biodistribution of transplanted cell. So today I will take you all to another kind of study and another type of pluripotent stem cells. And you can say that this is the original pluripotent stem cell which was known to humankind. And even the induced pluripotent stem cell to establish that they're pluripotent needs a positive control as human embryonic stem cells. So my group works majorly on the neuronal niche comprising of glial cells like astrocytes, oligodendrocytes, Schwann cells, and looking into their physiological function and into the pathological conditions such as in Parkinson's disease for the astrocytes. For the oligodendrocytes, we have selected the hypoxic ischemic encephalopathy and diabetic neuropathy for the Schwann cells. So here I am going to share our data and present our findings from one of my PhD students' work. So modeling in vitro neonatal hypoxic ischemic injury, significance of human embryonic stem cells and imaging methods. HIE, which is hypoxic ischemic encephalopathy, is a condition where severe brain injury occurs in neonates due to birth asphyxia, basically due to interruption of uterine or fetal blood flow and or hypoxic condition resulting due to failure to initiate and sustain respiration after birth. So hypoxia, as the word literally means lack of oxygen. Ischemia means restricted blood flow. So over here, the moment we say restricted blood flow, the key element which brings in the brain damage is glucose and encephalopathy affecting the brain. So perinatal asphyxia leading to HIE, coming to the global incidence, is a big burden in developing countries. Literally 26 births in every 1,000 births undergo HIE conditions. And as you can see from the cartwheel over here, that among the neonatal deaths, nearly 45% of the neonatal deaths are being brought about by perinatal cause and HIE. 20% of global incidence of cerebral palsy is due to this injury. So the etiologies of HIE can be due to three factors. That is well known: due to maternal reasons, due to cardiac arrest or asphyxiation or anaphylaxis or hypoglycemic shock, so on and so forth. And fetal reasons such as fetal-maternal hemorrhage or cardiac arrhythmia, and also uteroplacental conditions such as placental abruption or cord prolapse, etc. Now, coming to the severity, what we get to know from a study from India in 2017 is that most of the cases of HIE in India is in the mild and the moderate HIE. Why severity is important? Obviously, the long-term complications, the neurological and cognitive disabilities, such as seizures, mental retardation, and cerebral palsy, their intensity would be severe when the insult is severe, and it will be milder in case for the moderate and the mild cases of HIE. And of course, the lifespan will be more for the mild and the moderately affected HIE cases. But such infants, it is not that they are capable of having a complete normal cognition abilities eventually. So obviously it becomes a burden for the individual and for the rest of the individuals. And the underlying mechanism of injury and why endogenous repair is not happening is still not known. Coming to the existing treatments, well, the most practiced treatment, which is accepted worldwide, or rather I would say that with respect to its safety measures, is reducing the brain metabolic rate. And that's being done by hypothermia. But it's completely symptomatic intervention which goes in. It's not sufficient and effective to prevent long-term neurological and cognitive disabilities. Now, there are many compounds which can be suggested, which are used even in adult stroke conditions, but the side effects over here, because they are neonates, it's immense, especially because these compounds are known to affect the endocrine system of the infants. So the target endogenous repair mechanism, there aren't any compounds or strategies which are looking into it. The existing small molecules or compounds or pharmaceutical drugs which are present has severe side effects, and optimal regimen is just restricted to hypothermia. Now, initially, HI was largely considered to be the damage of the gray matter, quite obviously, because the neurological and cognitive abilities were getting affected. So it was first thought that the neuronal cells are the cells which are getting damaged and the gray matter is getting damaged. But fortunately, with the advent of new age imaging techniques such as magnetic resonance imaging, that's MRI, and diffusion imaging technique, we get to know that the HI has become as synonymous as white matter injury. So it very clearly and thankfully lets us know that, okay, in the brain, now we get to know it's not the gray matter which is getting most affected. The symptom might be through the neuronal cells, but at the end of the day, the loss which we get to find in the brain, in the developing brain, especially in the moderate and even in the mild cases, it is the white matter injury. So we go back to the developmental biology, the ontology clock during the neonatal birth. So why the white matter injury? To bring about a relevance, what we can see now with this figure and this map is that during neonatal stage, the neurogenesis continues to take place and the glyogenic clock is on. That's when the myelination gets initiated, and specifically the work for the oligodendrocytes and oligoprogenitor cells, they are coming into forefront over here at this particular time point. And indeed, through postmortem reports, we get to know that the OPCs and the oligodendrocytes, that's the OLs, these are the cells which are even less in the HI conditions. So unfortunately, while there is evidence suggesting that neurogenesis can occur, because we know that in neonatal, in the germinal region, there are neural stem cells, there are neural progenitors present over there, and they have, and it's very well known till the 5th year of our birth, the brain is very plastic and it is capable of repair. And because of the point of neurogenesis being happening, unlike in the adult brain. But we also get to know and that this endogenous repair mechanism is definitely then getting affected. Otherwise, why are we having to see a loss of the white matter? That is the loss of the OPCs and the oligodendrocytes. Again, when we go back to the postmortem report, did we find any report about the neural progenitors or the neural stem cells getting affected, the number getting affected? No. We find that neural progenitors and neural stem cells, rather they suggest that there are certain reports suggest that with hypoxic insult, there was an increase in number, but there was decrease in the OPCs and the OLs, and obviously the white matter. So what does our existing HI model state? There are numerous of rodent models which are being performed and carried out and reported. But in 2002, the Mouse Consortium Conference and that publication had shown very clearly that species difference in developmental clocks, specifically for the brain development, is very evident between the rodent and humans. So that makes the vulnerability, to interpret vulnerability and regenerative capacity of neural stem cells or neural progenitors difficult to interpret, and making obviously treatment interventions spooned. Coming to the in vitro work, what is present over there? Well, again, immense work is present with human neuroblastoma cell line, which is SHS555, even another secondary cell line, but that's of rat origin, PC12, and rodent origin primary neuronal cells, or even oligodendrocyte cells. So for the rodent origin, again, the same question comes over here, that how do we interpret the developmental clock and the regenerative capacity. And coming to the human origin SHSY55 cells, we don't yet know. So we will address that in our narration. So to correlate in vitro findings with clinical pathology, the first requirement is to establish an in vitro work. Why in vitro model? Because it is easier to recapitulate and understand cellular events and define what is going on in a single cell level when you are having the model in an in vitro platform rather than the whole body organisms. So there are major developmental differences between the rodent and the human brain we get to understand, is why it is making that we don't have yet an HI model in our hand of human origin. And not to blame, it is difficult to have human neural stem cells or neural progenitors, just like any other human neuroblastoma cell line, to be present in our lab to start out our work with, because we know it is inaccessible and it is ethical concerns are there. So it is just not possible to, even if we wish, to start out our work with human neural stem cells, and that too from the fetus. So why human origin in vitro model is needed? There is another bigger reason, global reason, what we can understand too. And I want to share that with you all. That despite increased knowledge of disease mechanism and higher levels of financial investment going on for research, for R&D, the number of novel medicines approved by regulatory agencies such as FDA is showing a steady decline. Moreover, on top of that, we see higher attrition rates of candidate drugs, which has been in the late preclinical development or in clinical trials. So is it, it's very stark and clear, because in science, we have to believe in facts, we have to believe in what is going on, and we have to get back to then understand, ask the proper question that whether we have to change the in vitro model to go ahead and look into what is going on wrong, in particular for human origin cells. And that too in the particular developmental clock. So for that, we took the help of stem cells. Just to give you all an introduction, I know that all of you all know the property, the two cardinal properties of stem cells are self-renewal and differentiation into different lineages. There are two major classifications of stem cells: adult tissue-derived stem cells, which shows limited self-renewal and multipotency, and the embryonic stem cells, which are isolated from the inner cell mass of the blastocyst, and they are capable of, they are immortal, so they're capable of innumerable self-renewal, and they're capable also to give rise to all three term lineage cells. So the other stem cell, which we also got to hear from the morning, are the iPSCs, but they are derived from the somatic cells after reprogramming, and we are trying to mimic the embryonic condition in the iPSCs. Now, advantage of HESCs than in vivo model for specifically for HIE is because the HESCs are being derived from the ICM, that is at the perfect developmental stage, and they're known, well known and capable of undergoing gastrulation and then to neurulation, and henceforth be it to giving rise to mature neuronal cells or to any type of glial cells. And hence, because it has been so widely and robustly proven, it is considered as a mandatory event to be performed, or a test to be performed by the FDA with respect to developmental toxicity. Then the other technique, which again gives an a tool or gears up our research, is live cell imaging and imaging per se. Why? Because anything we want to see in science, we are not happy just to see a qualitative research. We want a measure, we want a quantification so that it can be reproduced, it is robust. And life cell changes such as migration, say for neural stem cells, a migration or a proliferation, these are dynamic changes which are happening. So for those, we definitely need life cell imaging or imaging with a spatiotemporal pattern being present in our experimental design. Another reason why we go ahead for imaging even in our daily life when we are conducting our in vitro work. So a part of it for the pluripotent stem cells, you all got to know from yesterday from Aditi's talk that blue imaging comes very handy to recognize that, my starting cell type is pluripotent and they are embryonic stem cells or, iPSCs. So why is it important? Because for human embryonic stem cells, it has been also reported that if you passage it for long through enzymatic digestion, it can become pro-oncogenic. So you won't know whether it is due to the oncogenic genes getting upregulated or it is the true human embryonic stem cells. So we again over here, when we are passaging, we do manual passaging. And that too, how do we identify the colonies to be taken? We do the blue imaging to identify that. So definitely this is a tremendous boon for us because it is cost effective. You don't need to spend anything. You just need a fluorescence microscope in your biosafety hood and you can perform your regular day experiments. Now, the other reason why we had why imaging is a boon for us is we look into regularly and routinely the intracellular calcium dynamics, which we need imaging to understand at the single cell level. And even when we are using, say, WG-MSCs, that is Wharton-Stellia MSCs or dental pulp stem cell MSCs, and I need to state over here because there are many students who you all are present in the audience, you all should know that even if they're capable of getting passaged for n number of passages, say P12 and P13, we need, and the karyotype is normal, and the self-renewal seems to be normal, but we need to, for every passage, look into the formation of micronucleotides. So the micronucleus present in just beside the nucleus, the moment it starts appearing, we know that nuclear deformation has got initiated, even though the cells are proliferating. So this, again, another simple imaging technique needs to be inculcated in our regular day work to understand that the cells of interest we are working with are healthy and physiologically active. So for the present work, the aim of the study was to understand the influence of hypoxic ischemic injury on the vulnerability of human neural progenitors derived from human embryonic stem cells in reference to cell survival and oxidative stress, and assessing their cellular deregulation with respect to glutathione level, basal calcium, glutamate release, and intracellular calcium response to physiological stimuli. Beyond this, we were also interested to look into and evaluate the fundamental potential of neural progenitors, which is self-renewal and proliferation, neural and glial progenitor pool, and migration. So as I said, that evaluating at every stage is important. So we went ahead first to characterize the HESCs for the pluripotent markers. Then we derived the neural progenitors from the HESCs. And we had to prove that these neural progenitors are indeed neural progenitors through functional assay like differentiation to terminally differentiated neuronal cells, wherein they were electrophysiologically active, and also to glial cell type. The other cell which we took into consideration for our comparison was SHSY5Y, because this is the cell type which is extensively used till date for the work of HIE to understand cellular deregulation. And I want to show over here that SHSY5Y has a little different signature than our neural progenitors. Neural progenitors express the neural and the glial progenitor markers extensively, such as nestin, Musashi, A2B5, and S100 beta, whereas in SHSY5Y, we get the terminal neuronal markers, such as beta-tubulin 3, synaptophysin, tyrosine hydroxylase, then neurofilament, getting expressed. So definitely it's in a little more matured stage than the neural progenitors, which we are interested over here. And why are we interested? Because we want to know what happens wrong once you get the OGD, that is the oxygen glucose deprivation or the hypoxic ischemic injury to the neural progenitors. So because there are no study till yet, so we had to go to standardization of different percentages of oxygen, and also the time-dependent study of oxygen concentration. So we had used over here 2% oxygen instead of the 21%. And at different time points, we first monitored the cell survival and the ROS generation. What we saw that at the 90th minute onwards, the ROS generation was starkly clear in the neural progenitors, and it had started even in the SHSY5Y cells. But the cell death initiated from the 120th minute. So we chose the 90th minute as the time point for our OGD insult, that is oxygen glucose deprivation. Why? Because we wanted to represent through our in vitro work the living infants, the infants which have not been fatal due to the OGD insult or the HIE. So coming to the susceptibility that to further reactive oxygen species, we had taken H2O2 as an insult. Why? Because it's a byproduct of cellular metabolism. And what we found from the upper panel is that, again, we looked into the ROS generation and the cell survival, is that definitely the ROS generation, again, was very high in the neural progenitors in comparison to the SHSY5Y cells. But the difference in the toxicity, which was not evident in H2O2 in their normal self, when they were not undergone an HIE insult, has become pronounced when they were exposed to this H2O2 and they were exposed to OGD. So that's definitely indicating that there is something wrong about the antioxidant mechanism, or it is more vulnerable, the neural progenitors, than the secondary cell line. So we went ahead to check the glutathione level. And indeed, the glutathione level was, the reduced glutathione is what we measured over here, was significantly lesser than that what we found in the SHSY5Y cells. Now, glutathione and its derivatives can be even effluxed out through its transporter, MRP1. So the glutathione, the reduced glutathione level can be also due to it being getting effluxed out. So we had to measure the transporter MRP1 on the NPs and the SHSY as well. And we found that there was a decrease over here too for the transporter, thus indicating that the reduction in the reduced form of the glutathione in the neural progenitors are mainly because the glutathione is getting oxidized and not due to its efflux. ROS generation, or the basal ROS getting higher, is very intricately linked to basal calcium and glutamate accumulation or glutamate release. So hence, to understand the other pathological condition, that is the basal intracellular calcium levels, we went ahead to determine it using imaging technique. I will show further on the images and all. So we went ahead over here using the fluo4AM as our fluoroprobe. And as this graph indicates, there was a distinct increase in the basal calcium, but the trend throughout till here is similar in both cell types. It is a little higher in the neural progenitors, unlike in the secondary cell line. So here comes the interesting bit. When we started with the physiological stimuli to look into the spatiotemporal response at the single cell level of the neural progenitors and SHSY, what we found was that a complete opposite result was coming out. So at the control situation, wherein we have not given the insult, the SHSY5Y gave a higher influx of intracellular calcium when they were stimulated with KCL. That is quite expected because, the SHSY cells are more mature. They express the mature neuronal markers. So probably the number of mature voltage-gated channels expressed in neural progenitors are known to be lesser than mature neurons. So that can be the reason why we saw the difference. But what happened after the OGD treatment is that the neural progenitors showed a burst of increase, that is influx of calcium when they were stimulated with KCL, whereas in SHSY, we did not find the difference. The difference was negligible. So this was a very stark difference and an opposite phenomena and a dynamic cellular event, which we noted through imaging technique. I'll just show you some example. So in the left-hand side panel, it's the control neural progenitors where the response is coming. And here is the neural progenitors on the right-hand side panel, which had undergone the oxygen glucose deprivation. So it shows very, even visually, you can understand that the increase in intracellular calcium, which has been beautifully captured by the fluo 4AM dye within each cell. So each cell singularly, we can quantify and measure it with respect to time, and that too in milliseconds. So when we go ahead, so there is a huge coordination of channels and voltage-gated channels, which brings about a response to KCL stimulation. So among them, we screened which are the groups of channels which are known to be mostly affected by hypoxia, and they were termed to be the voltage-gated potassium channels. So there were numerous studies from smooth muscle cells, from artery cells, from cardiomyocytes, wherein they were said that the potassium channels are the ones which shows differential regulation when they were subjected to even just a hypoxia. Another complexity came in over here, that within those studies we could understand, and there were certain studies where there were comparative evaluation, where they showed that it is differentially regulated. So one cell type, if a certain voltage-gated channel got down-regulated, it was not the same phenomena being observed in the other cell type. So we obviously needed to screen this expression over here too. Again, for the trend was quite similar for the voltage-gated potassium channels, be it 1.5, 4.2, 4.3, the decrease being more stark and vivid for the neural progenitors than SHSY. But the channel which, the two-pore leak channel, potassium leak channel, gave a completely different data than that what we observed in the SHSY cells. So we found a distinct increase in the expression of task 1. Now, task 1, why it is required? Because that is the first player. So when you give KCl into your extracellular media, so over there, first, as per the electrochemical gradient, there would be intake of the potassium ions, which depolarizes, and hence it will open up the voltage-gated calcium channels, and that calcium entry would lead to the release of intracellular calcium from ER through ryanodine receptors. So task 1 is the first one, which ensures the response happens. Another thing I wanted to mention about the task 1 is that there are studies where it has been shown very clearly that with difference in pH of the media, of the cellular pH and oxygen content, the endocytosis or the expression can get affected. Next, we did with another physiological stimuli, which was ATP, because we were interested now to see that how it responds, dynamic response of these cells in comparison to the well-established model, which was SHSY. And here too, again, we find a very stark difference upon ATP stimulation when the cells were subjected to OGD. And when we get back to literature again, so there are pretty much quite many studies where they have shown that P2X and P2Y receptors can be regulated by oxygen levels, and obviously through pH too. So we went ahead to check over here the expression of the P2X and P2Y receptors. So coming down to the summary table, what we can understand that, yes, the difference in the control and versus OGD is very stark in the neural progenitor cells, but the main difference between the two models was evident and got initiated, or we wanted to report it because it was completely opposite response with respect to the intracellular calcium, and which was captured. It wouldn't have been possible for us to understand that if we wouldn't have had the live cell imaging facility. So in sum, our data suggested that SHSY cells cannot adequately substitute the human neural progenitors for developing in vitro culture of HIE. So we had done some more confirmatory tests, whether using inhibitors for task1 and inhibitors for the P2X and the P2Y receptors and IP3 receptors to check whether the response is decreased or not in the OGD subjected neural progenitors. Now, coming to the cardinal properties of neural progenitors, the first property is they should be able to proliferate. They should be expressing self-renewal property. So that was being met through three kinds of experiments. One was colony forming unit assay, then neurosphere formation assay, and cell proliferation protein Ki67 was detected. And what we get to find is the clonogenic and the proliferative potential of the human NPs at the till 90 minutes of OGD insult did not affect. Now, coming to the glial and the neural progenitor markers, what we found was the nestin did not get affected, but definitely we found that there was loss of glial progenitor markers in the neural progenitors. Coming to another very important property of neural stem cells, which is migration. So if it is not capable of migrating from the germinal layer to the place of interest where it needs to go and give rise to oligodendrocytes and bring about myelination, then it's of no use. Even if we have a number of NSCs, it's of no use over there. So indeed, BFGF dependent PSA and CAM, which is known to be the primary requirement for the migration of neuroblast is getting affected. The expression of the PSA and CAM goes down, and even for FGF, it goes down. Now, over here, there can be a question that we see a decrease in FGF, then why isn't it that the proliferation is getting affected? Well, we found a corresponding increase of EGF, and it is known that FGF and EGF works in a compensatory method for bringing about an self-renewal and proliferation. So probably due to that reason, we did not find, because there was an increase in EGF receptor expression, we didn't find a difference in the proliferation. So concluding from this part of the work, we can definitely say that yes, indeed, the neural progenitors derived from human embryonic stem cells gives the opportunity to us to track them and set them at the proper developmental stage. And it has advantage than the secondary cell line because the intricate details, the cell function is completely different, and it is a totally different ballgame in the human neural progenitors than when we are comparing it with the SHSY5Y cells. So human embryonic stem cell derived NPs definitely should be taken for serving the purpose of modeling HIE targets or any of the property to be evaluated. We further, this part of the work got published in Stem Cells in Development last year. And we further went ahead to work that, so there is a reperfusion phase after the OGD. So what happens in the reperfusion, as the terminology literally says, that back again, we get the oxygen and the glucose. So that's what is being provided even in the culture. So our next question was, okay, fine, we saw that during the deprivation phase, when the insult was there for 90 minutes, now what happens? I'm giving them back their oxygen and glucose. So is it that they're able to recover because do they show the regenerative potential over there? So the same set of experiments were performed, and we had taken, you will be seeing over here, WJ being written over there. So this strategy was also being seen whether to evaluate the neuroprotective effect of non-contact method of WJ MSCs on these neural progenitors. But I'll share only few slides with you all to show that indeed in the reperfusion phase 2, even though when they had got back their oxygen and glucose, the task1 expression, neither the task1 expression went down, that was permanently showing higher expression, And the KCL response was still high than the control neural progenitors. So we can also take the task one as now a viable biomarker even, which is starting to show the difference from the OGD step itself. Now coming to the what happens to the proliferation story now and to the neural glial progenitor marker, do they get back to shape and are they worsening or they becoming better? Well, the proliferation we find here that it is starting to decrease, and it showed a significant decrease than the control. And the neural and glial progenitor marker expression over there, the neural progenitor marker did not show any difference. Again, the S100 beta and the A2B5, the oligoprogenitors and the astroprogenitor marker, they were definitely lowered. Similarly, for the migration, there was no betterment with respect to migration, even with the reperfused cells. And so over here, what we can see, I've just given the summary wherein you can see the first panel with the yellow boxes, they represent the control. Next, it is the OGD with reperfusion, that in all these parameters, it is going, the deformities and the degeneration is starting out, the deregulation is starting out, and pretty much of them getting repaired or recovered when we are providing the WJMSC conditioned media or co-culturing them through transfell system. Well, I would like to show over here in the last panel that even with the WJMSC conditioned media, we had found that there is a down regulation of task1 expression, which was not seen in the OGD reperfused cells. So the reason being, it is the growth factors are known to work through the tyrosine kinase receptors. So through downstream PI3 kinase and through PLC gamma, it is capable of regulating the endocytosis of task1. And that is the probable reason why we find the task1 expression down-regulated in the neural progenitors, which has undergone that injury, but we have provided the WGMSCs as a treatment strategy. Such are the cases for, so to do the other experiments too, that's why I used the inhibitor for the PI3 kinase to show that whether the WJMSC repair mechanism is actually PI3 kinase is playing a major role for it or not. So that part in details I have not gone for, but that's precisely what the fourth panel over there shows. So in conclusion over here, what we find that the cellular susceptibility of the neural progenitors in the reperfusion phase is definitely not improving, and WJMSCs through non-contact method can bring about significant improvement. Why is it improvement, the non-contact method? Because in cell-free mode, it is capable of providing the niche support. This is much more user-friendly than giving the cells themselves. So this part of the work got published in Cellular and Molecular Neurobiology. And the litmus test of whether these OGD reperfused cells are getting affected to give rise to oligoprogenitor cells, oligodendrocyte cells eventually, that even we had performed. I'll just make you all go through a few of the data points which we had got. So from the neural progenitors, they were subjected then to oligoprogenitors and eventually matured to oligodendrocytes. What we found over here that definitively the neural progenitors which were subjected to the insult of OGD and reperfusion showed distinctly lesser number of oligodendrocytes. And similarly over here too, we tracked the task1 expression, and we had found over here too the task1 expression remains increased in the OGD insulted cells. And coming to the other progenitors at the OPC stage, the O1 and O4 and E2B5, they are definitely downregulated in the insulted cells. And this is qualitatively to see some of the markers of O1 and O4 at the OPC stage. On the right-hand side panel, there are the control OGD plus reperfused cells, and then the OGD plus RWJ in the presence of the Wharton's jelly conditioned media, wherein we can see that there is a partial recovery, and that was being quantified by flow cytometry. On the histogram on the third panel, you all can see that definitively there is a partial recovery and a good amount of O1 and O4 recovery when in the presence of WGMSC conditioned media. The endpoint at oligodendrocyte matured state, again, over here, the MBP was evaluated through immunofluorescence and through flow cytometry. And we found that definitively the recovery in the presence of WGMSC conditioned media was near to control, unlike that in the insulted cells. So we went ahead to check that whether these oligodendrocytes, which we have differentiated from the pluripotent stem cells, are capable of producing myelin binding protein, and whether which part of the neuronal cell it is capable of enwrapping and making the myelin or coating it with it. So we took pure neuronal culture, which is NSC34, and we labeled them with PKH26 dye. And we further did an immunofluorescence of these cells with MBP, and there was no expression of MBP. They were just expressing the red color dye, the PKH26. So when we co-cultured them with the oligodendrocytes, so once they matured in the co-culture, and we went ahead for labeling them with the MBP protein, we saw that near the neurites is the place where the MBP expression was quite prominent. So we went ahead to do a dual marker expression through flow cytometry. And what we found was definitely this association was very detrimental or very impaired in the OGD reperfused neural progenitor cells, unlike the control. And this recovery was definitely present when we had given the conditioned media from the WGMSCs. Further, we had also done electron microscopy. So one panel I've just put over here to show that there were mitochondrial defects in the mature oligodendrocytes. So I won't take further on, y'all pull through it more. So the present work basically shows that the neural progenitors, which undergoes the OGD insult, that is what happens during the asphyxiation, it shows prematuration at the OPC stage, and thus resulting in decrease in the myelin binding protein at the matured stage. It also shows decrease in association with neurons in co-culture. Thus, obviously, the myelination of the neurons will be getting impaired. And WGMSC CM exposure in the reperfusion stage, it restored these impairments, suggesting that paracrine action of WGMSCs was definitely beneficial. Significance of this study, well, in vitro model got developed from human embryonic stem cells, and we didn't limit it to the neural progenitors and their cellular dysfunction and the dysregulation, including many dynamic functional studies, which we did. Beyond that, we pulled it to the OPC stage and to the mature oligodendrocytes. So I remember yesterday one of you had asked whether glial cells can be generated from pluripotent stem cells. Indeed, it can be generated. And if you follow the developmental cues very beautifully, even the phases can be looked through and be obtained. And we have done it with the classical pluripotent stem cells, human embryonic stem cell. And I would mention over here, this is Indian origin human embryonic stem cell bought from Reliance. So considering that developmental toxicity is a mandatory test by FDA on HESCs, so our present model can definitely serve as a cell-based platform to evaluate therapeutic strategies targeting any developmental stage which involves neural progenitors or oligodendrocyte progenitors and oligodendrocytes. So I would like to acknowledge the funding agencies who were related with this part of the work. So CSR had funded this work, and my PhD student, she has just now received her degree. She was funded for a fellowship from ICMR, and NIMHANS definitely for the infrastructure and everything. Thank you. inside. No, here only. Look here. Where is it? Okay, sorry, there's some. Yeah, I'll take the questions. So the first question is there a genetic component to HIE? No. Per se, with respect, it is a physical injury which occurs during that particular time point. So during birth. So that's the reason why the oxygen and the glucose deprivation occurs. Does HIE result in autism? Yes. There are reasons that there's a wide umbrella of cognitive disabilities which can occur due to this insult. And the effect of less presence of myelin and the white matter in autism is also related. So definitely it comes, it even spans the umbrella of autism. If yes, then you think using ASD specific, patient specific iPSC line be a better disease model? Yes. So I had this question even from the reviewers and the examiner. So we have healthy iPSCs, but in particular, we wanted to go for the developmental stage from HESCs because, as you know, it is widely accepted that the developmental clock is best mimicked by the human embryonic stem cells because as simple as they are being derived from the inner cell mass. So once we start with the iPSCs, we have to initially show the neuronal and the clock, the neuronal and the glyogenic clock in the iPSCs, whether they're mimicking the HESCs or not. Eventually, if for the ASD in particular, so in ASD, there are certain genetic components too. If those genetic components has to be taken into consideration, then definitely the ASD specific iPSCs can be worked into. Because NP is only a component of the disease pathology. Also in future, do you plan to look in organoid? Yes, definitely we would. We are very much interested to look into organoid culture to understand that in total, what happens to the total developmental stage when specifically we are looking into in a neonate time point. So it becomes very important. Thanks a lot. Can we? If there are any more questions, I would be happy to answer. Okay, with that, we will, I thank all of you to be here and today from time. So it was smoothly conducted within time point. So we will take a break for lunch now, all of us, and please log in again to join with the advanced microscopic techniques, which we will be starting from 2 p.m. onwards. We would like to request all of you, please try to log in at least 10 minutes prior so that we can start out the talk at 2 p.m. onwards. Thanks a lot.
test. Hello? Can you hear me?
Yes, we
Ah,
can hear
cool.
you.
Can you see my screen?
Yes.
Ah, perfect. Okay, so now I try my camera and
Yeah.
then I'm done.
Yeah, then we'll wait for some time.
Yeah, so can you see me?
Yes. Perfect.
Okay, perfect. Nice.
So,
It seems to work.
yeah.
So then hear you later. Okay?
Yes. Shadi, bye.
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 Hello.
Hello. I'm just checking the number of participants. So we'll wait for a few more minutes, at least 5 minutes.
Sure.
To agree.
Mhm.
Because it's they're going to come back from lunch. So
Yes,
it's a little
I
challenging
can
for
imagine. Yeah. Very important.
you. Yeah. Good afternoon, everyone. I can see now participants have again started logging in. So I'm sure more will be logging in. By that time, let me initiate the last session of our symposium. So the theme of the symposium for the last session is advances in microscopy. So we will be listening over here advances, be it in confocal microscopy or in electron microscopy and cryo-electron microscopy. So our first speaker is Dr. Oliver Schlicker. He's a senior application manager in Leica from Germany. So Oliver is a neuronal cell biologist by training. And after his PhD, he was heading an imaging facility in Stuttgart, Germany. He has a strong background in confocal and wide field microscopy and image processing and analysis, and joined Leica Microsystems in 2008 as application manager, now responsible for advanced fluorescence wide field microscopy. So with this, I would hand over this presentation area to Dr. Oliver.
Thank you very much for this nice introduction. And thank you for inviting me for giving a presentation to this great symposium you are heading here. So I say hello to everybody who is joining the session. And yeah, so the topic of the session is about advanced wide field microscopy. And I especially want to go into one of our, let's say, actual products we are doing in Leica Microsystems from the wide field business, which is called the Thunder Imager Live Cell. And I would like to present you in this presentation, especially also how you can combine these modules or this system with photo manipulation and total internal reflection fluorescence microscopy with the Leica Infinity modules. So again, thank you very much for inviting me, and let's start with the presentation. I hope you can hear me loud and clear. Otherwise, just tell me if anything doesn't work very well, so I can react on that. Okay.
Yeah.
So let's start with the first part of it, which is about the Thunder Imager itself, what it is and what it can deliver. Well, Thunder Imagers is a new class of systems we offer from the wide field side within Leica Microsystems, which can reduce the time to come to result very much. I will explain what that means in the next few slides, but also they can extend the range of applications you can do with the wide field microscopes dramatically, as well as they can also increase the relevance of your data itself, because within wide field, we have a typical problem, which you can see in the first image and in this slide, which shows a tumor spheroid, actually, which contains a lot of cells, which has a rather thick shape and so on. And if you image it with a wide field microscope, you directly see the inherent problem of such samples on a wide field microscope. And this is the blur, which masks all the wanted information, and therefore it makes typically thicker samples difficult to image with conventional wide field microscopes. So that's why we invented the Thunder technology. And with Thunder, you get from the same image this information. And you directly can see that from this image, you now have a much better contrast, and you can easily see much more details in this image itself. So therefore, Thunder, besides other things, also removes the blur and therefore makes thick samples accessible for analysis and any other things you would like to do to get numbers out of an image later on. So here's another example on a already thundered image. It's actually a TIE scan of a fixed gonad of a C. elegans. And actually, we take a look now at this position here, which is marked with this dotted frame around there. And the customer, actually Professor Eckmann in Germany, in Halle-Wittenberg, they would like just basically to know how many nuclei they can see in the different positions in the gonads of the C. elegans at the first thing, okay? And if they would use just a wide field image and they would run, for example, a 3D analysis out of that, because of the inherent blur where you have a lot of nuclei available, all of the light will overlap, and therefore it makes it nearly impossible to resolve later on in the analysis the single nuclei. And therefore you get a wrong number at the end if you just do a basic 3D analysis of that, because here you have a lot of light which is overlapping, and therefore you cannot really distinguish between the different nuclei in here. Here, where you have much less available, then it's really nicely possible, but here not. So take a look at this number, and the total number of detected nuclei in here by this image was 156 by doing this type of analysis in there on the raw data. So if we go on and we use the Thunder data of that coming from a Thunder microscope, you nicely can distribute the single nuclei in this part of the gonads of the C. elegans. And now we have a number of 1186 in total in the same field of view and in the same spot we are looking at, just by using the right amount of raw data in the analysis. So here you can see that getting the right data for analysis is very important at the end to get the perfect data out of it. But I'm sure you know that in your daily business that you have to work on images basically after you did the acquisition itself. I will come to this point a little bit later on again, but also with Thunder, the way how we create the systems, how we put components together like light sources, microscopes, objectives, as well as stages and cameras. This is perfectly fitting together so that by using just less light to get the same image, you minimize the light doses to your sample, especially important for living samples, because all of us know that the more light you put on your sample, the more you interact with the sample, and let's say the higher the chance is that you introduce phototoxicity to your sample. Another very important point in using Thunder images is that you increase the statistical significance by, for example, acquiring more data in less time, but also correcting the images, especially for the background and the inherent blur, which are typically available in wide field images. Here you see one example what I'm talking about. This is, for example, a whole mount retina from a mouse from a customer in Murcia in Spain. And they, of course, use a Thunder imager. And with the Thunder imager, you always get also the raw data if you want. And this you see here in the upper row. In the lower row, you see the real-time data you get with the Thunder imager available. You directly can imagine that without the inherent background you see around the structures, it is much easier to quantify later on these structures. Here you see another example, also from this customer in Spain. Here you see especially the retinal ganglion cells marked in red. And as this slice was rather thick, you see also all the out of focus information of other cells laying in planes below the frame or above the frame. And therefore, this is also overlapping. And if you just would like to know the total, or let's say the real intensity level of each point here in this image, it is rather hard to distinguish, or rather hard to compare points in the darker area with points in the brighter area. Here you see an example on how a line scan should look like. The peaks are the corresponding spots underneath this line here. And here we see the intensity level, and the x-axis is actually the length of this line. So if you would like, for example, compare these peaks with that, you think, okay, they rather have the same intensity, but you have to imagine that the total intensity of the spot is basically a sum of different components. First of all, it is the intensity of the spot itself at that point in this focus position, but on top of that, there comes also all the out of focus information, which is also adding to each and every point in the image together. So that means if I have here a higher background, but a lower total lower intensity of the spot itself, the signal might be the same, or the total signal might be the same as this spot where I have a little bit lower background and so on. So this is very important to mention here. So with Thunder, you directly get data like that. So first of all, on the image, it looks much more contrasty. And this is mainly because the background is gone, so to say. Yeah, so you have a much lower background, a much better flat field of the image itself. And therefore, you now can start to compare the different peaks here now together. Now, these two peaks, which we compared in the upper image, which looks the same intensity, they have different intensities, for example. And now you can imagine how important it is to have the real data for quantification. But I'm sure you know that. Okay, coming to the physiological relevance of your data, I mean, this highly depends on the components you're using to acquire these images, to keep the cells alive and happy, or to all your samples you're imaging. So here, with every Thunder imager, we combine all the components necessary to have a higher physiological relevance during your acquisition of the data already. And this comes because, for example, of a real-time controlled and high-speed acquisition possibility. And also because of the higher sensitivity of the system itself by combining the right light sources, right objectives, but also high-end SCMOS cameras with it to get as much as possible data out from the optical system. And to keep the sample alive over a long-term period, we also equipped the system with climate equipment for temperature control, CO2 control, so for buffering the system, and also O2 control if you want to also perform hypoxia experiments, for example. So with these components, we also, and with Thunder, we also reduce the time to the result. So why is this? Why do you say that? Well, in a typical classical wide field workflow, you typically start to acquire an image set, a time lapse, for example. Then after this time lapse image set is finished, then you typically go to post-processing process to prepare your images, as we have seen before, for example, to get the real analysis data out of your image. And this is actually at minimum a 3-step process. Typically, this post-processing processes contain different types of processes which need to be done to get an analysis data. With every Thunder imager and the integrated software workflow, during the acquisition itself, all the images are prepared as you have seen before, so to get the higher contrasty images, and they are automatically shifted to the analysis workflow so that at the end, after your acquisition is finished, you already get the analyzed data. So what does that mean? Well, here we have a very basic first starting video on how it looks like in the LSX, our software, our imaging software. So we have here two channels of a fluorescence sample here. And to activate Thunder in the image workflow, you just need to open this window and just select one of the three different methods, which I will explain in the next few slides. So here we just select a method called instant computational clearing, which is the fastest and the most effective method here. And directly after the images have been acquired, by just clicking the acquire button, we directly get the result. And not only that, you always, and per default, the system is set that it always keeps also the raw data available, which is using for live calculation of this result. So what you get is, if you like, always the raw data, so you can always go a step back, and then also, if you like, also post-process with other Thunder methods, but you directly can apply the Thunder methods directly to the acquisition workflow. This works also for stat stacking and for any other, excuse me, for any other imaging process. So for example, here we acquire a live stat stack, and during this acquiring the stat stack, you can directly see the Thundered results, so to say. Another example here, a tie scan in multi-channel with fast acquisition of 3 channels here over this whole kidney slice here. And you directly can switch to the live Thunder view, so to say. So during this acquiring the 3-channel image data, you see here the single tides flying in this tie scan, you directly see already the Thundered results. But as you see here in this list, you always keep the raw data available if you like. You can also deactivate that. Now, what are the different Thunder methods and how do they compare to each other? Well, as you have seen in one of the videos just before, there are actually 3 different Thunder methods you as a customer can decide to use during the acquisition. Okay, so the first method, it's called instant computational clearing, depicted by this lightning bolt here. Okay, the second method, it's called small volume computational clearing, which is depicted by this schematic drawing of a neuron or of a cell, so to say. So this is mainly for small volumes used. And then we have a method called large volume computational clearing, which is mainly for thicker specimen like these spheroids I showed you, or organoids, or even bigger specimen like C. elegans or other model organisms. Okay, so Thunder itself is a method which we call is an optodigital method that removes the autofocus blur which is inherent in wide field data in real time. It is, as you have seen in the images and in the videos, fully integrated into the system's data stream. So you just activate it and the result is this. And this is very important, it automatically takes all the relevant optical parameters which are needed to calculate this type of image on the fly into account. So every time you switch an objective, Thunder knows it. Every time you use a different camera setting, Thunder knows it. Or if you use a different RAW4, Thunder takes it into account, because this is important to calculate the real data of the wide field data out of it. Okay, so in the next few slides, I will come a little bit more close to what's going on during Thunder itself. And this is a more or less schematic view on the technology itself. So let's take a look at the inherent problem of a wide field microscope. Well, you have an optical system which is imaging, let's assume an infinite small spot. So a very small spot is imaged through this optical system. And what you get out of it in the image is not, again, a spot because of the fraction of the spot which is happening when light is traveling through the optical system, you get a point spread function. So a distribution of light into the spatial area around the spot itself on the image side. So which looks, for example, like that. This depends a little bit on the type of objective you're using. So now imagine you have a sample on the microscope itself, and the sample is consisting thousands and millions of such spots which are overlapping. You can imagine that all the spots creating a point spread function with a shape like that, which is much bigger, and are overlapping, you will get a blur in the image, which you see here as a crop out of a fluorescence image here on the right side. So with Thunder, especially with Thunder instant computational clearing, we detect, let's say, features in the image by knowing, okay, how big is a typical feature when we use a certain type of objective and a certain illumination method. And then we reduce the local blur around these features, which you can describe as we just get rid of any overlapping or any disturbing light around a point spread function itself. So at the end, we have smaller point spread functions, if you like, with less stray light around. And therefore, you have less stray light and blur in the sample, which enhances the contrast. And this is a very, let's say, basic method per se, but detecting the features takes let's say you have to know where the features are and how big they are. And as we know what optical components we are using and how we are illuminating, we can calculate that. Okay, another method which I described to you is the small volume computational clearing. Small volume computational clearing works basically as a 3D deconvolution, like you know, for example, from Richardson-Lucy 3D deconvolution methods. So it's a described method, but Leica especially is adding a special adaptive process to detect noise, which is also adding to the images in these three-dimensional volumes we have here. And to detect noise, we're using a technology which is called decision mask. The decision mask technology was already introduced for our confocal microscopes some 2 or 3 years backwards, and they are using the decision mask technology in their lightning approach. Yeah, so lightning is the brother of Thunder, if you like. So the decision mask technology combined together with a 3D deconvolution reshapes a point spread function in a wide field microscope so that it reassigns wrongly assigned photons here into wrong planes into their place of origin. So at the end, you get a much smaller and brighter point spread function. So a much better dot, which is more referring to the original as you see here. And therefore you see much more details and with a much better signal to noise ratio in the image. So of course, this is better than the ICC because here we're reassigning the photons here. But I will come back to that example in bigger or in thicker samples. This is very much true in thin samples where we have a nice where you can nicely distinguish between the different point spread functions in different Z planes. The last method here is the so-called large volume computational clearing. So what it does is it combines both methods together. It combines the instant computational clearing and it combines the small volume computational clearing in a single, or let's say in a two-step process, but you just select this method here and the software is automatically doing that during the acquisition. So what it does is it does out from an image set from wide field, it does first of all the instant computational clearing calculation. And then by using a different point spread function, which is now different, together with the 3D deconvolution and the decision mask, it reshapes the point spread function and you get a clearer image compared to the ICC. Now, as you can see, as you already have lost some information in thin samples here, the effect is not as good as if you directly use the small volume computational clearing in thin samples. Yeah, I will come to that back later to that. What happens if we have really thick samples where you can nearly not distinguish between any structures, like you can see in here, the white field. So now coming a little bit to some words about the decision mask itself. So the decision mask is actually a map of signal to noise ratios over the image. This is done by using a kind of a kernel, which is scanning through an image, like an example image you have here. We have structures which are very bright, so which have a higher signal to noise ratio, and darker structures like here, which have a lower signal to noise ratio. And this is typical in an image. In an image, you have high variations of signal to noise ratios, and deconvolution typically is highly dependent on, let's say, a higher signal to noise ratio. If you have a low signal to noise ratio, then it's a problem. So actually, what we get is a map like this. So the red data here shows region of high signal to noise ratio, and the blue colors here show regions of low signal to noise ratio, as I have already mentioned in the images. Well, this means if we have a high signal to noise ratio, we can do a different approach in doing a deconvolution, because the chance that we deconvolve a real signal here is much higher than in these regions where we have a low signal to noise ratio, where the noise is really high. So what does it mean if we have a region with a high noise level and a low signal to noise ratio? You can see that in one of these diagrams here. So where we have, so our eyes immediately can see, okay, it's noisy, but there is a peak inside. Yeah, so intensity over position, a line scan we have here. It's very noisy, but our eyes immediately can see that this is a peak. However, a deconvolution algorithm typically uses a global approach, and therefore it typically, for example, adapts to a high signal to noise ratio. But this low signal to noise ratio levels here might be seen as, or the noise in this peak here might be seen as real signals. So that means it might happen if you don't have any regularization of the deconvolution process, then you might get wrong deconvolutions into the noise. Yeah, which doesn't really enhance the signal to noise level at the end. So this doesn't really help. So now we need to have a regularization, which is based on this map you have seen. Red is low regularization, and blue with high, with low signal to noise ratio needs a high regularization in the example we see here, for example. If you do that, then the algorithm detects mainly this peak as our eyes does, and therefore the deconvolution is happening so that it detects the noise and therefore only works on this peak here. So, and the result is this yellowish line here. So we get a much better signal to noise ratio in this yellow line by adapting the regularization factor in it. This, you as a customer don't need to care about that, because automatically, if you select the small volume computational clearing or large volume computational clearing, the software which is running there is taking care on it. Okay, so now let's have a example on what's going on a thick sample. And to make a schematic drawing here is, we have lots of point spread functions which are overlapping in our, let's say, object. If the object is imaged through our objective and through our microscope, we have a lot of point spread functions which are overlapping, which results in a very bad signal to background, or let's say a very bad contrast in the wide field. You all know that if you have a thick sample and a high numerical aperture objective, you get not much real data out of the wide field microscope. With instant computational clearing, we use now this method by detecting a structure, also, for example, here in this region here. And then we are, let's say, deleting the background, the local background around these different structures. A schematic drawing on how the point threat functions would look like here, you now can distinguish between them very much or much better. And the result in the image is already a high enhancement of contrast of this image, where we start to see smaller structures in it. However, it looks like as it could be a little bit better. Okay, so now let's take a look at the small volume computational clearing, which is the 3D deconvolution with the decision mask. Well, as I have said, if you have, of course, noise in, but also a high overlap of point threat functions, a basic 3D deconvolution algorithm fails typically on most of the structures in here because it is very hard to distinguish if a photon from here belongs to this point threat function or belongs to this one. So what you get is wrong reassignment of the photons and therefore not a real enhancement of the image quality itself. You get a little bit better contrast, but number of structures, for example, these lines in here is not there, right? So even not with the small volume computational clearing, yeah, because it's basically a 3D deconvolution for thin samples. But however, if we now compare instant computational clearing by making the point threat functions now available for a upfollowing 3D deconvolution, which we do here, then we can gain more contrast and more structures out of this image, out from the wide field image. Remember, this is the raw image we have, and this is the final image we get by using large volume computational clearing with thick data sets. By the way, this was a crop out of a brain. So the sample was a brain slice with 160 micrometer thickness, and it was imaged with a 63x oil objective with 1.4 numerical aperture. So very, very low optical or a very high optical resolution, and therefore a lot of blur in the wide field image, but still structures like these little processes can be visualized by using large volume computational clearing. So with all these methods and with the Thunder Imager, we now can enhance the field where we can use wide field microscopes. Yeah. So typically wide field microscopes could be used in the past, mainly for 2D cell cultures like this, so thin structures, maybe a little bit 3D cell cultures, and combining it with basic 3D deconvolution. But now with all these technologies, we now can enhance the field of where we can use wide field microscopes also to bigger samples, and therefore close the gap towards confocal microscopy, for example. Okay, for different methods, we have different Thunder imagers, and we call it Thunder imager family. For model organism imager, where we need to have a big field of view and a big travel distance from the objective to the sample and so on, and you want to prepare and interact with the model organisms, for example, we have the Thunder imager model organism based on a stereo microscope. For all of those customers who want to use mainly slices or slides on an upright microscopes, we deliver Thunder imager tissue or 3D tissue here based on upright microscopes. For customers or users who want to use basically living samples in a cultured dish, multi-well plates, something like that, we have a system range based on inverted microscopes, which are called Thunder Imager 3D Assay, 3D Cell Culture, or Live Cell. They depend a little bit on the configuration. So the live cell, as the name says, comes with an incubation system to keep the cells alive. The newest family member is also the Thunder Imager EM CryoClam, which combines our correlation light electron microscopy feature with the shuttle here to put it then later on a scanning electron microscope and have free samples underneath the microscope, but also pre-screen the sample with a Thunder imager. This is our newest family member. However, in this presentation, I would like to go a little bit further into live cell imaging with 3D cell cultures or live cells, and then also doing later on photo manipulation and membrane imaging with it. Okay, so that's why I would like to concentrate on the Thunder Imager 3D cell culture and 3D live cell. So again, here's some images of the 3D assay, our basic model, always equipped with a scanning stage and a high sensitive camera, then with a faster stage and a more sensitive camera and more features of the microscope coming with the 3D Thunder Imager 3D cell culture. And then where we are concentrating now is the Thunder Imager live cell, so the fully live cell equipped instrument for live cell imaging. And then later on, we also add components on the Thunder Imager live cell for photo manipulation and turf. This will be the last part then of my presentation. All Thunder Imager live cell, they share always the same components to enhance the sensitivity overall of the system to gain or to get less phototoxicity during the experiments. And this comes because of the combination of Leica LED light sources and our real-time controller to control all of the components together in one sequence. In combination with the high sensitive CMOS cameras and specific lenses, which are gaining more light out from the sample, we have a complete system to gain sensitivity over our experiments. So how does, for example, a real-time controller really helps to lower phototoxicity? Well, if you have just a PC and running and controlling all different hardware, it's typically controlling the hardware sequentially and typically not as fast as if you would have a real piece of hardware controlling directly each and every component. So with a PC, for example, you might have the problem that the illumination needs to be longer on than the real exposure time of the camera, because not the two things like switching on the light source and controlling the camera are not perfectly synchronized, because the PC is running also the software, other programs together. So the synchronization is not really perfect. So if you have a real-time controller, which is inbuilt the microscope itself, and it knows from each components in the microscope, so for example, camera, light source, and stage, then each component is directly synchronized to each other. So in our example with the exposure time and the illumination is only as long as the exposure time of the camera needs it and not longer. So therefore you save a lot of light already, but get the same image. So very important for live cell imaging. And therefore it minimizes the light exposure, photo damage, and bleaching over that. And this comes together also with the control of our LED light source and also the flexibility of the LED light source. Here you see an example of one of them we can offer. This is our LED 8, and the LED 8 shares up to 8 different LED lines to have a really broad flexibility for different fluorophores and also nicely switching and combining different LEDs during the experiment. Then these components come also in game with our adaptive focus control, our focus holding mechanism to keep the sample in focus without illuminating it over it. So therefore you save also more light, because you don't need to have an focusing let's say a focus step by acquiring images in different planes, but still keeping the cells in focus. Then last but not least, we have an integrated fast moving stage. We call it the quantum stage, which is a linear motor stage, which is directly integrated into the sequencer control of the microscope to enhance the speed of acquisition together with the light sources and the cameras together. Here you see an example of a multi-well plate on the stage where we acquire 96 wells, 1 position per well, 10 set planes per well, and in 3 channels, and we can acquire the whole plate in less than 3 minutes and 30 seconds. Just to give you an example how fast this can acquire. Yeah. So, however, besides these components, you can add more to it. So one component we can add is a specified module for turf imaging. I will come to turf imaging in the next slide. This is the component you just can add to the module, and it contains an automated turf setup and enhancing the possibilities of the system very much. Together with a laser light source, which can hold up to 5 different lasers into one place, which you can use then for turf imaging. And of course, the microscope and the software, you have an automated turf setup for imaging, membrane imaging, and so on. So what is TIRF? TIRF stands for Total Internal Reflection Fluorescence Microscopy, and it is an effect caused by refraction between the glass and the water surface. Here you see an example where a laser beam is reflected, total internal reflected after it reaches a certain angle from the surface here. And with the help of that, you excite mainly fluorophores at the glass-water interface by the so-called evanescent excitation field. So you have total reflection of the laser beam, and some energy is traveling into the sample itself and is exciting the fluorophores very close to the glass membrane. To get images like that, so this is a typical wide field image, and then to switching on TIRF, you only see the fluorophores available at the glass-water interface here. Another example you can see here. So this is, for example, a actin GFP expressing COS cell in a cell culture, and you nicely can see the actin fibers close to the cover slip shown in red, and other things which are not anymore close at the cover slip are then darker visible. You see the nice movement of this growth of this lamellipodia here in the beginning here, or the retraction of the cell, the growing of the cell at different sites, very nicely visible within TIRF methods. So why do we need TIRF? Well, because we need to know, for example, what's going on in the membrane of a cell. So with typical wide field microscopes, you, as I explained already a lot before, you excite the cell totally, so to say. But with TIRF, as we reduce the position where we excite the cell only to the glass water interface where the cell sits on, we can now take a look at protein movements in the plasma membrane, for example. Yeah, or plasma membrane dynamics and so on. And TIRF can be also used for biophysics. So for example, here an in vitro experiment with, for example, motor proteins in a solution and adherent microtubule fibers, and you only want to see the motor proteins attached to the microtubule fibers. In wide field, you see low contrast because you have a lot of unbound structures available. But if you switch to TIRF, you see nicely only the motors at the microtubule motors, as you can see here in a real example. So the Leica Infinity TIRF looks like that from the box. So it's a complete motorized box which you can add, which comes with different features, like, for example, our TIRF scanner, which controls the laser beam automatically, and a so-called collimator, which focuses the laser beam automatically. So this is just the hardware to really easy have a TIRF setup later on to do that. Then we have a so-called merge optics in, because you can combine epifluorescent imaging for standard fluorescence imaging with TIRF imaging together over a single path. Then we have the so-called beam expander, which is then enhancing the light density in in the sample itself to use the TÜV module also for super resolution imaging. And then the crucial part within the TÜV module is the so-called TÜV sensor, because with this TÜV sensor in this position here, we detect whenever the laser beam is total internal reflected from the sample and then traveling back into the system. Well, this is looking like that. So when you do a so-called auto alignment to help you to set up the real perfect TÜV for your sample, is you just click an auto alignment button. What the system is doing is it's illuminating the sample and it is changing the incident angle of the laser beam until it gets a total reflected laser beam detected by the sensor. When it is detected by the sensor, it knows which type of angle we have with a certain color of the laser beam. And out from that, it can further calculate also the refractive index on the sample. All of this belongs together by the so-called Snell's law. And this refractive index also defines then with different colors what TÜV angle we need to get a certain penetration depth. Penetration depth is highly dependent on also the TÜV angle. So it sounds very complicated. You as a user just click the auto alignment button. The system automatically aligns itself. And then you as a user, you just can reproduce always the same penetration depth by, for example, clicking on 110 nanometers. And dependent on the wavelengths you're using, it is automatically adapting the TÜV angle. So with a method called the expert mode, you have also a free combination of setting up the laser into different penetration depths. So we always calculate the penetration depth, not the angle. The angle is just how it's using it. But you as a user always select the penetration depth here by this slider. And this, as you can see, the slider has two different zones, a green zone and a red zone, a green zone and a gray zone. And the gray zone here is the so-called halo mode, where I come to that a little bit later. And you have this 360 circle thing where you can change the azimuth itself. What does azimuth mean? Because of maybe dirt in the sample or something like that, you have different contrasting appearances depending on which angle or which position the laser beam is illuminating the sample. So you might have got interference artifacts in an image at a certain position, but if you change to a different azimuth, you get a nice flat TÜV image. Halo means highly inclined laminated optical sheet. And this is a method where you're just not in wide field illumination and also not in TÜV illumination. So we're above the critical angle and you're illuminating mainly the sample. So this can be combined with TÜV imaging by just changing between two different channels. So for example, the green actin channel and then the red lysosome channel we see here in this channel here. Lysosomes are typically above the plasma membrane here, and therefore need to have a different illumination method during an experiment, but you nicely can combine it. With TÜV, we also deliver the right objective, actually a set of 3 different objectives, which always share a correction ring for different temperatures and also for different cover glass thickness. And all objectives also have a very flat field of view. That means we correct up to 19 millimeter field of view, which is very important because TÜV is a very flat method. So if you have a flat field of view, you nicely can also use also the big field of view. Compared to non-plan corrected objectives, you might also see. With the 160x objective and the possibility to use the beam expander in the so-called high power mode, you can also perform high resolution or super resolution experiments with it to gain data like that. So it is using the same module, but just a single button click and a different objective. You can use the same module for doing super resolution microscopy by doing the localization microscopy principle known as from our GSD or ground state depletion system. or storm, you can also say. So what is using here? It switches off all the fluorophores by putting a lot of power into the sample until the fluorophores are in a so-called dark state, which is very long lasting, and then they fall back and they form this blinking events. So only stochastically, some of the fluorophores inside this image here are on. And so therefore they're timely separated and therefore can nicely being fitted by a fitting algorithm. And out from that, by acquiring a number of images here, they automatically can calculate a super resolution image with this method here with the TURF module. Its brother from the brother from the TURF module is the so-called infinity scanner. It's actually a galvo scanner which guides a laser beam throughout the sample, where you can, of course, can write something into that, but its main purpose is photo manipulation. And why do we need photo manipulation? Well, typically you want to see a reaction on cells, but cells or samples are typically, if you just let them grow, in a steady state. So they're lazy, lying around. And what you need to do is you have to have a tool to wake them up or to manipulate them. A tool here in our example could be this, but our tool is the infinity scanner itself, which is a galvo scanner having a laser beam together with a fiber switch to switch between TURF and scanner illumination, a type of software, and a different type of laser for doing ablation and so on is also can be equipped. So this specific laser we call the pulse laser unit is mainly used then for cutting, ablation, and introducing DNA damage or uncaging experiment. So with all of these different methods in the scanner, for example, we now have a big field of applications we can share for detecting protein or for measuring protein dynamics by using FRAP experiments, protein interaction with FRET acceptor bleaching, sample simulation in optogenetics and uncaging, or introducing sample damage. Because of the just the different wavelengths we can activate, we can combine all of these different applications with the help of the scanner into one, let's say, tool, okay? The scanner comes with also features like a specific aperture diaphragm where it can also reduce the light and first of all, the light which you put on the sample, but mainly also change the thickness of the spot how you're scanning. Why this is the case? Well, if you have a small spot, you have to scan more lines for covering a full field of view here, or let's say a region of interest. But if you have a bigger spot, you can use less light. But a bigger spot means more light for a bigger area, but that means also then also less light on the spot. So everything together can share also different or can have different effects on the samples. So you have a lot of variances which you can use then for the scanner by changing, for example, the scan speed or by changing the power density by the numerical aperture. So that's why you can use the scanner for different types of applications because of the wavelengths, but also because of the tools we offer to change the spot and the light intensities. What is crucial for photo manipulation is also doing photo manipulation in the same position as you're looking at or as your fluorophores are. And this is because we offer also the Vario optics, which is an optical tool to refocus UV light, for example, so into the same plane as your visible light is also at, yeah, so that you manipulate the same position as you're looking at. And with the help of these different features, we can also use different regions of interest with different shapes, but also with different intensities, which can be controlled nicely by the software, where you are freely you're completely free, so to say, to set up different types of experiments, combining different imaging methods with thunder, without thunder, with different types of regions of interest, with loops and so on. So it's a freely definable wizard where you can define different experiments for that. Last but not least, I would like also to come to some of the typical applications, and let's start with our thunder imager with photo manipulation. And here we have two examples where we, for example, photo switch a green, let's say a Dentra dye, which has a green fluorescence first, and by photo switching using a 4 or 5 nanometer laser in different shapes, you can switch it to the red position. And here next to it is a photo activation and DNA damage experiment by just illuminating a line by using a 4 or 5 nanometer laser, and then simultaneously by introducing the photo damage, you also photo activate photo activatable GFP, which is here attached to an histone H2B in that case. Yeah, and what you see is if photo damage sites appear here, these two lines move apart from each other because here DNA is distracted, and therefore repair sites happening in this position, and the speed on that is then measured at the end. Here we see an example where we photo activate a different photo switch at 2 different positions by using photo activatable or photo switchable actin. Another example shows a typical FRAP experiment of fluorescence recovery after photo bleaching at this position or here. Then we have here a flip experiment of fluorescence loss in photo bleaching, where you keep a region constantly bleached and you measure how the drop of the intensity is in a different region. And this gives you any information how the distribution speed is between these 2 different regions, for example. Here you see actually a combined experiment where we see a single frame with 2 cells. One cell is performing a FRAP experiment here, where already you have seen the recovery. I started again. So here we have a FRAP, fast recovery, and here in this region, we keep it constantly bleached to remove the unpolymerized tubulin in this lower cell, which enhances just the contrast for the turf image, by the way. So this is also a turf image scenery. And then here we have an example of ablation of cytoskeleton in MDCK cells. Here, for example, cutting of a stress fiber in the cell here, nicely seen by using the PLU. And here we are cutting a cell-cell connection by using the pulsed laser unit. Yeah. And then last but not least, you can also use it for even cut cell walls. So it's very strong, and it's even as strong that you can mark glass for micro-patterning, or you can activate micro-patterns by using the Infinity scanner in this example. And here is the combined experiment of Thunder Imager live cell with turf, and then we activated the different Thunder methods. So you nicely can see that the raw white field data looks like that. If we switch to turf, then it looks like that, already a better contrast. Turf and Thunder ICC already enhances the contrast, and the best result you get with Thunder large volume computational clearing. Then we have here an ablation experiment where we destroyed little vesicles here of cells expressing GFP MX1 in MDCK cells. And then you see also the corresponding Thunder image data coming out from the system like that. Some other nice examples are here, just 6 samples, side-by-side comparison of the raw data you get and then the direct data you get out from the Thunder Imager. Also here, a 3D reconstruction of an organoid cluster or cortical neurons in microfluidics, neuronal 3D cell cultures as a side-by-side comparison in 3-dimensional reconstruction. Yeah, I just go on. And with that, I would like to thank you, and I'm open for questions. I already saw that we have questions in the chat.
Yes. Then I think you can go ahead and take one by one the questions from the chat.
So one question was how Thunder works while imaging the cell imaging the cell biomaterial interfaces, especially to analyze the 3D proliferated cells on an assumed thickness. Well, it is so that you typically set up an experiment like you would do with any wide field microscope. So for example, you would do a set stack over different positions. We have there a tool which is called a navigator, where you perform, first of all, a nice overview of your sample. And then you select by just clicking the different sites which are interesting for you. And then the system, you can define a set stack, and then the system keeps the focus stable over the different positions, acquires the data, and by just activating Thunder to this, for example, Thunder instant computational clearing, you directly get already the raw data. The system knows by your setup how thick your sample is. It knows from the objective itself and the illumination you need what type of wavelengths you're using and what through you are using. So you typically set that automatically up. And then this information is then used to calculate the real data. But always don't forget, we keep the raw data in case you want later on to also run it through a different process. So during the acquisition, you start, for example, with ICC, and later on you want to try out the large volume computational clearing. If you find out that is the better choice, directly you can start during the acquisition with large volume computational clearing in the next experiment. Okay, so the next question is, in case of tumor spheroids grown on 96 well plate, how will Thunder work without any interference? Well, it's exactly the same. We always recommend to have 96 well plates with a glass bottom, because if you want to have the highest image quality. Okay, so the highest image quality you get with a glass bottom with an immersion objective. Sometimes if you need to go deep into a sample, an immersion objective has not enough working distance. So then also specific working distance objectives, which you then can also use. So all the bunch of different objectives are used. So the best would be if you contact one of our local sales colleagues, of my sales colleagues, and you discuss with them what your typical application is so that you get the right optical components for that. Thunder always takes care on the different components. Yeah. So, and then, yeah, so if you have plastic dishes, it will also work, but you have to use different objectives for that. So no problem at all. Then just came in a last question. As compared to confocal, what is the efficiency of the software? The thing is that the software is really comparable. So if you know the Stellaris software running, the confocal software running, you directly, let's say you need 15 minutes to find around or to find your way how to set up an experiment in wide field software. The same is true on an stereo microscope or on the CryoClam software or on our upright microscope software. Everything looks the same. It just depends on how much wizards you have. One example you have seen in one of the videos how the software is set up. I hope that answers the question. So it's very comparable. Yeah.
Thank you, Dr. Oliver. You and this more and precise. So our next, we will move on to our next.
Okay.
And here will be.
Thank you very much, and I wish you bye-bye.
So the next talk, I'm sure you all are all holding your breath to listen to because it's one of a kind, been there in our country, and it is headed by so Dr. K.R. Vinod Kumar is a tenured scientist at National Center for Biological Sciences and in charge for the National Electron Cryomicroscopy Facility. After his bachelor's at Madurai Kamaraj University, he joined the School of Biotechnology to complete his master's in the field of biotechnology. He earned his PhD in membrane protein biochemistry and biophysics, primarily using cryo-electron microscopy technique from the Max Planck Institute of Biophysics in Frankfurt. After receiving the coveted International Max Planck Research School PhD Studentship Award in the year 2001. After his PhD, he worked further with Dr. Richard Henderson at the MRC Laboratory of Molecular Biology, Cambridge, as a Marie Curie and MRC Career Developmental Fellow, where he trained himself in both X-ray crystallography and cryo-electron microscopy. With that introduction, I would request Dr. Vinod to take on the presentation and start it.
Okay. Thank you, Dr. Indrani. I thought I will give an overview of electron cryomicroscopy.
Yes, surely.
Yeah. So I am at NCBS, but the campus we are, it's called the Bangalore Life Science Cluster. It has NCBS InSTEM, which is another DBT funded institute, and CCAM, where there's a lot of startups are hosted. And today I thought I will give an overview of how cryo-EM has developed, and it comes very nicely after Oliver's talk on light microscopy. So I will explain how we are able to now reach very high resolution of single molecules, which are purified protein or DNA protein complex, and then how the field is going towards in situ, which is probably more relevant to the current symposium. So why use electrons? So you can see here from the spectrum here that there's a lot of different wavelengths, and a 300 kV electron, which is the commonly used one, although people use 100 and 200 kV. So you have a wavelength of about 0.02 angstrom. So this is very small. So it's possible to get very, very high resolution, but there are issues. So we generally don't get this high resolution. So resolution is one thing, but there are other problems. So here is just a table to compare between light and electron microscope. So you have radiation source, which is light and electron. The wavelength which is used, even for 100 kV electrons, we have about 0.04 angstrom, the wavelength of the source. And instead of glass lenses, we use electromagnetic lenses. And one of the biggest disadvantages is that you have to use vacuum, which means that you cannot observe live cells, which you can do with light microscopy, like Oliver mentioned. You can combine both light and EM, which I will briefly mention in the end of the talk. So within electron microscope, there are two ways one can image specimen. You can have scanning or transmission electron microscopy. So in SEM, which is scanning electron microscopy, the image is formed by detection of reflected or knocked off electrons. And in TEM, electron pass through the sample to create an image. Generally, a projection is created. And because they are 2D projections, we need to reconstruct them into forming a 3D volume. So the information obtained in SEM is about the sample surface and composition. And the way the images are formed in SEM is that you have a set of coils which scan the beam like in a raster-like pattern to collect electrons. And as I mentioned, the resolution in SEM is lower than in TEM. And if you have a very expensive aberration-corrected transmission electron microscope, at least in material science, you can reach a resolution of less than an angstrom, as high as about 0.5. In SEM, the sample thickness is not an issue, but in TEM, it is an issue because electrons scatter a lot. And so if you need to have something like 500 to 600 nanometers or less, and if you want to get very high resolution, then it has to be even thinner. And typically we have 80 to 300 kilovolts as the voltage for TEM, and in SEM 2 to 30 kV, and the magnification here is higher. It's easier to use in SEM, but for TEM in particular to do cryo, there's a lot of steps and training needed. This is just to show for the flavor, because we are still going away from the pandemic. These are the SARS coronavirus. This is an SEM image just showing just the round object. But when you do the transmission electron microscope, you start seeing the different shapes of the coronavirus and the spikes protruding out. And this images show how, and when we had this kind of a setup, you had to have in a special dark room and make sure that when you are imaging, the room is not disturbed or kept quiet. However, with this now, this enclosure gives a lot of stability, and you can work in light because the control is not next to the microscope but in a separate room, and that makes it much more easier, and this allows data collection for a longer period. So electrons see electrostatic potential. So you have an atom, you have nucleus and electrons surrounding the nucleus. There are different different cells of electrons. So when electrons interact with atoms, they interact with everything. When they interact with the outer cell electrons, there is low angle scattering. When they interact with inner cell electrons, you have medium angle scattering and high angle scattering with nucleus. So this is in contrast with X-rays or neutrons. X-rays interact mostly with electrons and neutrons interact with the nucleus. So because they interact very strongly with the material or the sample, what it means is that there is a limit on thickness. So in materials, for example, typically they aim for about 500 angstrom. If you have a heavy element, like for example, when semiconductor industry, they look at materials and they need to have very thin sample. Because biology doesn't have a lot of heavy elements, we can go a little bit higher and we can get away with this particular problem. And we get a lot of signals. So electrons interact with atoms like in different ways, but much of the beam, about 80%, doesn't do anything. It just passes through. Then there are about 5% of the electrons which scatter elastically. What it means is that they interact with the sample, but they don't lose any energy. There's a small change in the phases, but there is no loss of energy. And there is about 15% of electrons which scatter inelastically. And these are the ones which actually lose energy and they cause radiation damage. So elastically scattered electrons maintains energy and the inelastic scattered electrons create radiation damage. And in TEM, these are the ones which contribute to the images. And in SEM, most of these backscattered electrons and secondary electrons, they contribute to the imaging. In life sciences, if you have a microscope like this or any voltage, there are multiple things one can do. So you can look at molecules as single entities. For example, here there is a viral core particle without any DNA. This is hepatitis B virus core. You can see individual viral particles here. And this, you can then collect a lot of images to make a 3D reconstruction. Or you can also look at specimens which are helical, for example, actin or microtubule or flagellum. This is an example of acetylcholine receptor which can be made to form a tubular crystals. And using helical image processing, then you can get a structure like this. It's also possible to look at crystals. Here are thin 2D crystals of bacteriorhodopsin, but currently it's also possible to look at thin 3D crystals and use electron diffraction to get also high resolution. And the last is to look at the whole cells or sections of cells and then do tomography. Then it's possible to look at, for example, in this case, from my former colleague, you can look at the bacterial cytoskeleton in bacteria without either of these steps where you try to purify protein. So in the current state, there's a lot of possibilities what one can do with cryo-EM. For a single particle, the size limit is about 50 kilodalton now, and the resolution is between 2 to 3 angstrom, and Some of the very good examples, the resolution has gone beyond 2. Current limit is that less than 40 kilodalton, they cannot be oriented reliably. You can see them. So if you put a small molecule like lysozyme, you can see it on the images, but it's not possible to orient them reliably because of the signal to noise. As I make these talks, I keep changing this number because as time goes by, this number goes down. And in 2D crystallography or helical reconstruction, the size is not a limit, but in both these cases, you need to get well-ordered helices or crystals, and this can be very time-consuming. So in tomography, uh, the raw tomograms are very noisy because we use very low dose. You don't want to create a lot of damage. So as such, the tomograms are not very high resolution. But if you have objects which you can see in the tomogram, then you can do subtomogram averaging and get very high resolution as high as 3.5. I put here not high throughput yet, meaning most of these steps here are automated, meaning you can put a specimen, data collection is automated, processing is automated. In this case, this is still being developed, and in the near future, this also will become highly automated. And if you had a thin 3D crystals, you can do microelectron diffraction, and these are crystals which you will not be able to see by light microscopy, but once you put in the EM, you can find. And here also you can get very high resolution. So these are current possibilities, but I want to give an overview on how we came about here. So what are the technical advances that has allowed us to do a lot with the electron microscopy? So this is based on a review myself and Richard Henderson wrote in 2016. Let's start with, there are a lot of people, a lot of ideas, and a lot of technical advances. This has been for the past 40 to 50 years. And in terms of instrumentation, one of the main thing is how we freeze or how we make specimen. That's called plunge freezing. This was developed by Jacques Dubussy in 1982s. And in terms of instrumentation, microscope technologies, the field emission gun, which is the source of electrons, has made it much more coherent. The beam is more coherent, and this makes a big difference in comparison to what we had as a tungsten filament. Then the cold stages where we put our specimen has become very stable. And to keep these samples at liquid nitrogen temperature, you need to have a very good vacuum, and this has been made very, very good. And then the way we have been looking at images has started with films. Then we had CCD. Now we have direct electron detectors, which are based on CMOS technology. And similarly, computation started with cross correlation. Now we have maximum likelihood. There are many more programs are being now developed. Most of them are using now machine learning, and things are getting really fast. And because of advances in GPU through video games, most of this processing, which used to take weeks and months, now can be done in hours. So the earliest biological samples looked by EM started with Helmut Ruska, who is the brother of Ernst Ruska, the person who made the first microscope. Helmut Ruska was a doctor. He looked at samples which are air dried. So here is a bacterium, and these are the phages. So these are not stained, not frozen, just air dried, and one could look at things like this. And then also people started looking at a lot of viruses, and here is another virus which is air dried. The next step came by introduction of what we call negative staining. So in this, you take a sample of your interest, put it on a EM grid, then apply a heavy metal like uranyl acetate or phosphotungstate that forms an envelope around the protein molecule or the macromolecule. So it doesn't stain the molecule, but forms an envelope, which is why it's called negative staining. And then you start seeing objects like this. So this was introduced by Sidney Brenner in 1959. This is a TMB image which he did. And this already gave very nice images. So these are textbook images. You open any textbook, you can find them. And using the negative staining method, Aaron Klug and David de Rosier, they developed the first programs for making three-dimensional structures from projections. So this is a T4 bacteriophage, and what David De Rossi and Aaron Klug did was to apply uh, program or algorithms to do reconstruction from this projection. And one of the reasons why Aaron Klug was awarded a Nobel Prize for formulating this conversion of projection to 3D structures. So this was a helical specimen which they did, and subsequently this was the same kind of principle was then used for icosahedral viruses by Tony Crowther in 1970s. So these are different viral particles which are shown, and what Tony Crowther got for tobacco bushi stent virus here. So people realize that using negative stain is good. You can look at specimen. It has limitations because the grain size of the metal, the heavy metal use or the stain use determines the resolution, which most often cannot go beyond 20 angstrom. And people were trying to see how they can overcome this. And this is when Bob Glaser and Ken Taylor at UC Berkeley, they devised a way to freeze samples. And this is one diffraction image of catalase crystals. And this was the first time somebody had done a frozen sample in TEM, and they could show that the crystals were preserved and you can get spots very nicely. And in the meantime, Jacques Dubussy at EMBL Heidelberg, he and his group also were studying water and trying to figure out how to make a very good specimen by converting water into ice, which doesn't form crystals. So this is from a review, which is a very nice one from Jacques Dubussy, which shows how they developed the plunge freezing mechanism or the whole protocol. So the first instrument they used was just a rod with a rubber band, and then a forceps where you had the EM grid, and then the ethane, which is here in this cylinder, or you can also see here, you can see the EM grid here. All they did was to put a sample, for example, even water is enough. They removed the excess fluid, plunge frozen in ethane. Then these are the things they studied. You can see that there are three different types of ice, which is again shown in this review. So these are hexagonal ice, which you can see from this diffraction pattern. And if you heat this sample, you get cubic ice, which can be seen here. And what we really need in looking at different life science sample is to have amorphous ice, which is shown here, which doesn't give any pattern. And you can see the beads which they used here. So this process was developed by them, and they also had to develop various means to also keep the frozen sample in the microscope. So they developed, I think, the first holders and the cryo blades to keep the sample cold, and also the importance of vacuum. So typically in a lab, what we do is, this is a plunge freezing apparatus. You have a rod, and then you have a forceps where you have a EM grid. And this is the chamber where we are freezing, and you have liquid nitrogen, and this inner cylinder has a liquid ethane. Liquid ethane is used because of faster, higher freezing rate. And once you apply the sample, you remove the excess fluid, and then you freeze here in ethane. When you look in the TEM, this is how a grid looks. And we have holes here, and this is carbon, and these are holes. And the hope is that when you blot and you freeze in ethane, there's a thin film of ice which is formed. And in that ice, we are hoping that we have our biological samples. And this is what we image then. And this was the first, one of the first few images that Jacques Dubussy and his group image. This is of adenovirus. You can see that this went into the cover picture of Nature in 1984, because this was the first time somebody could do a cryo image of a biological sample. And already you can see a lot of features of the virus. However, it took about 7 more years for them to get something reasonable in terms of a 3D reconstruction from this projection. That's simply because of computing and also the image quality and so on. The same virus now has been done by the group of Hangzhou in UCLA in 2010 using similar specimen preparation sample preparation, and they were able to get to about 3.6 angstrom, where you can build the whole virus and you can see here the density and all the side chains and so on. So this is a development that has happened, and it was realized by Richard in already 1995 that there are three parameters which limit the resolution attainable of biological specimen by EM. One is radiation damage. This is something which we cannot avoid because it's going to happen because electrons interact with the samples. But you can slow down by using liquid nitrogen or freezing. And we have, when the electron beam hits the sample, we have movement and also charging. The last one is how we detect. So in the last, I would say about 8 years, we have had tremendous progress made in this particular field, in all these things, but in particular to the detectors. So in the old days, when I was still a student, we used to use film as the medium for detecting. So these are in the microscope. So when you take an image, the electrons hit the film, which is coated with silver halides. And what we are doing is then we take the film, go to the darkroom, develop it, and then hopefully after developing, we find that the images are good or bad. So this was the one which we were using long ago. Then came the charge coupled device or CCDs, where electrons are converted to photons and back to electrons. These two were useful, but they had limitations because they were slow. Like in the case of film, you had to develop until you knew if the image was good or bad. And they had lower DQE, what we call signal to noise ratio for both these. Then there was a lot of emphasis put on making better detectors. And now we have what we call direct electron detectors because they detect the electrons directly, unlike film where it uses silver to detect. So in this is based on complementary metal oxide detectors or CMOS technology, where you have a epi layer, which is a sensitive layer which detects electrons. And then you have a passivation layer where you have diodes. So when electrons hits, it discharges some of the energy which is detected by this diode. And this substrate is forming the backbone or a support to the whole thing. So this is a 300 kV electron simulation where it shows that it can also backscatter. So this was done by my former colleague Greg McMullen as a simulation. So the advantage of these detectors is that we have a higher signal to noise ratio, and then they also operate in a rolling shutter mode, meaning they allow recording of movies. So this has become very important in trying to get high resolution. So I will explain in the next few slides what these detectors are helping us. So this just a graph to show the comparison of different detectors. So we had film, which had a very low DQE. Now some of the best detectors, you can see that the DQE, which is a measure of signal to noise ratio, is higher at lower frequency, and also they have reasonable DQE even at higher spatial frequency. So an image taken with a direct electron detector looks like this. So this is a single summed image, and there is about 25 electrons here. But this image is composed or made of 33 frames. What I mean by is that this is summed image, but because we record in a movie mode, so we have subset of this image, or in other words, the total electron was about say 30 electrons, but each of these frames during the exposure has been captured. So we can go back and ask what has happened during the exposure. So these movies add up to give this summed image. And by going back to see what has happened during the exposure, we can learn quite a bit. So one of the things which we learned was that from single frame, maybe we are able to see, for example, here the edge of the carbon. And if you increase the dose, then you can also start seeing the particles. by doing this averaging of the frames, it is possible now to follow what has happened during the exposure. So here the viral particles are marked in green and the background in red. Then you can see that during the exposure, uh, something has happened like movement, and this movement can be tracked over the frames, and this can be corrected computationally. Now we have several algorithms that do that, this motion tracking, and this can be corrected later in the by softwares. So by analyzing these kind of movie frames, what we realized was that uh, when the sample has not seen electrons, it's thought to be very good, but what us and others found was that the first two electrons, which are supposed to be very good, were actually not that good. These are B factor, which tells the quality of the images, and here is the electrons, the number of frames, and you have here in this axis you have the resolution. You can see that the first few frames are bad, then you have a plateau where things settle down and you have very high resolution as seen here, and then you have a decay here. So this decay is understandable because it's radiation damage. After about 15 electrons, you are starting to see damage. Damage meaning you have breaking of bonds and much more happening into the sample. Because we know what is happening, this also can be incorporated into algorithm. Um, and then what one can do is to, starting from a summed image, you can align, which is for correcting the beam induced movement, and also waiting for the later frames on the first frames, it's possible to get very high resolution. For example, here in the case of this hepatitis B viral core, using just less than 1000 particles, this is only 2 hours of work, you are able to get to less than 4 angstrom, and you get 3D reconstruction like this. And if it's in this movie, you can see that the alpha helices of these viral core particles are very resolved. You can also start seeing the side chain. So this you can do in a reasonably short time, and also because the programs have got better, the processing is also easier. Okay, so this is on single particle, and using our facility, we have had many structures, and some of them are published, some of them are about to publish. And what I will, in the next 5 to 10 minutes, I will go on is to explain what is possible and what we can look in the future, in particular for cells. So currently we can look at labile large complexes by single particle EM, in particular, a lot of eukaryotic macromolecules, membrane proteins. And one of the good thing about cryo-EM is that we can call multiple structural states. That's because you have a protein in solution, then you freeze the sample. So if the protein or the macromolecule is in a different functional state, then you can capture them and separate them by computation. And also, we can also get high resolution structure of actin and microtubules, and in particular, amyloid fibrils, which people now have started even looking from patient samples also. And just to end this part, I would like to highlight that the highest resolution obtained by single particle EM now is about 1.25 angstrom on a very well behaved sample by 2 of these groups. And so it looks like that if you have a well-behaved sample, you are able to get very high resolution of macromolecules. So that was single particle. So now we come to cryotomography. One of the things I mentioned was that if you had a thick specimen, then it becomes difficult with electron microscopy. So here are an example of a bacteria. So you can see here, this is a low mag image, and you can see that these dark objects are the bacteria here. So if you can go to one of these bacteria, and then you can image them at higher mag. So these are plunge frozen, and you can clearly see here now that there are 2 membranes, and if you are very keen eyes, then you can also see the peptidoglycan layer. So this is now possible, but however, when you look inside here, where the cytoplasm of the bacteria is, then there is not much visible or very clear because it's dense and also it scatters a lot. So this puts a limitation on what thickness one can look at look in TEM. And sample thickness of up to 600 nanometer can be looked. But if you want to get high resolution, then it has to be thinner. I put here 150, but other people also think that 200 to 300 nanometers would also be possible. So what do we do for thicker specimen? Let's say that you have a cell or you want to look at a virus replicating in a cell. So there are two ways. One what we call conventional way. You have a specimen, let's say a cell's tissue sample. The conventional way is to chemically fix, for example, by glutaraldehyde and then stain with osmium. You dehydrate in different steps, then embed in a plastic section it, and then you can stain the section again with heavy metal. So this is a conventional way where then you take it to a EM and then you either image one section or multiple of these stained sections and to get a 3D reconstruction. The other way is to immobilize the specimen using cryo methods. So as we saw, you can also use a plunge freezing to do this immobilization. But here the limitation is that you can't have anything thicker than about 1 micron. That's because the sample is very thick, and then if you try to plunge freeze, you will get crystals again because the rate of freezing will be lower, and you can't freeze greater than that. So instead of plunge freezing, it's possible to do high pressure freezing. This is where it's possible to freeze up to 300 micron thick samples in the smallest dimension. So what happens here is that you have a sample which is held together with the metal plates, and at about 2000 to 2400 psi, you have a thin jet of nitrogen being sprayed, and at high pressure, you freeze so fast that the water is not possible to form crystals. So you can also do freeze substitute and then embed in plastic, or you can directly go either to cryotomography or cryosectioning. All these end up in transmission electron microscope later. So just to show an example, if you do a conventional resin embedding method, this is of a bacteria, both are gram-negative bacteria, and this is of cryo freezing. This is called CMOVIS, cryo-EMO vitreous section. So you can clearly see here the difference in quality of the images, what you get from conventional embedding, and here you can see the two membranes and again the peptidoglycan layer, and in this case also the nucleoid or the DNA in the bacteria. So cryopreservation is better. It's most likely it will give high resolution, but it's much more demanding in terms of technicality. So either way, you go to a TM, you have, for example, when you make a lot of sections by conventional embedding, you take these sections, take an image, then you have a series of images of different sections which are aligned, and then you get a model. Alternatively, you can take one of the sections or even the frozen bacteria, take it to a TEM, do a tomography where you tilt the stage to get different views of the sample. Again, do stacking and alignment, then you do reconstruction to get a very nice model. So this is using TEM. There is another alternative way, which is what people generally call a volume microscope, where instead of using TEM, one uses an SEM. Here, the sample preparation is the same. You start with a bulk sample, like for example, a tissue or an organ. You go through these steps of fixation, dehydration, etc., and embedding in epoxy. So once you get a rigid block, then there are multiple ways one can do. So you can put it in a SEM, which also has a diamond knife. This is called serial block phase imaging, and where the diamond knife removes the or cuts the sample, and the SEM takes an image, and this goes on in a cycle. Then you get series of images. So instead of diamond knife, it's also possible to use a ion beam, which is typically used is gallium, which instead of knife also removes the sample from this block here, then you take images. So these are two ways, and the disadvantage is that the sample, once it is removed, you cannot retrieve back, meaning once it's, for example, this knife cuts, the section is gone after imaging. So alternative to this method is what is called ATOM, or automated tape collecting ultramicrotome. Here you have a knife which is stationary, you cut the samples in a defined thickness. Then on a silicon wafer, you have all these sections, which are then scanned by SEM, and you get again series of images. The advantage is that this can be stored, and one can look at it at a later period of time. So once you get all these images, these are series of images. These are really big because, for example, you can run this serial block phase imaging for a week with a big volume. Then you stitch and align, you get a big volume. Or in this case, for example, you take a brain and you can get all sort of neurons and things, and then you interpret. So this is where I think the whole field is going towards. And in particular, this last step is where there's a lot of emphasis being put on, because interpretation of these large volumes does take a lot of computation, and also algorithm is needed. So this is volume microscopy. This is not TEM, but let's go back to TEM. So in electron cryotomography, unlike where you take only projections, so here you have a sample. You start with the 0 degree, then you tilt the stage by 2 or 3 degrees, depending on the sample, and it goes all the way to plus 60, and then come back, and then you go in the other direction to minus 60. So this is called a tilt series, and typically one tends to get 20 to 30 micrographs. So this tilt series is then aligned because when you rotate the stage, there will be some small rotation and translation. And these are aligned using gold markers, or we call it fiducials. And once you align them, you can reconstruct them to get a tomogram. So here are examples where the gold particles are used for alignment. So the same section, this is from a tomogram. After reconstruction, you can clearly see this is of mitochondria. You can see the inner and outer membrane and the cristae here. So this is very powerful because you are looking at real things as they are in the cell. And people also have started looking at or modeling things inside. For example, here is a spiroplasma. These are very thin bacteria. Once you get a tomogram, then you can give, for example, this is a ribosome, this is a chaperone, and these are enzymes. And then these as a templates, and ask the computer to find where these are in this particular tomogram. And for example, in the case of ribosome, it works very nicely. You can see all of them mapped here by cross correlation. So once you have a tomogram, these sort of things are possible. And currently, the buzzword is called focused ion beam milling. This is now becoming very popular. What happens is that when you have a grid, you can grow, for example, a neuron on a grid, on a goal grid. You vitrify this sample by plunge freezing. Then using a gallium ion, you remove from the top and the bottom lot of sample or a lot of material. Then you are left with something what we call a lamellae. So these are very thin, and you can go up to about in nanometers. Then you do tomography of the same lamellae. So this is how lamellae look. These are TM images of chlamydomonas, which is about 100 nanometer in thick, and these are different yeast cells, about 250 nanometer in thickness. So because this ion beam allows to remove material, this can be now taken to cryo EM, and tomography can be done. And again, here shown different samples here. We have bacteria or archaea, which are tiny. You can do ion beam milling to get these wedges. And once you put in the EM, you can start seeing very nice features. This is applicable also to mammalian cells, where this is the lamella, which you can take it to the EM. And this is a mitochondria from yeast, where you can start seeing nice features. So this focus ion beam milling now allows what people call windows into the cell, where combining light and EM, then you can possibly try to localize the region of your interest, cut using ion beam, and then take it to EM. So this example is from this review by Grant Jensen's lab, where this is a Caulobacter. They wanted to look at one of this chemoreceptor array, which they fluorescently label. And here is a bacterium, fluorescently labeled. You can look at them, then take it to EM, do a tomogram, and then this localized this area where the chemoreceptor array is there, you can do subtomogram averaging to get a pseudoatomic structure. So the principle of subtomogram averaging is very similar to what we use in single particle. You have a tomogram, you know where the objects are, they are randomly oriented, you have a reference, you align them, and then you do iteratively to get averaging to get high resolution. And by doing this subtomogram averaging, there's a lot of structures now of different macromolecules within the cells. So these are structures not from purified samples, but from objects which are from tomograms of cells or cell sections like ribosome on ER or nuclear pore complex and so on. So this is a growing field, and there's a lot of new developments that has happened, and you can possibly see that there will be more and more structures coming from by just using in situ, combining both Fib and tomography to get high resolution. So in the future, this is what I think is going to happen, is to combine, or what we call integrative structural and cell biology, where you combine light microscopy with super resolution, transfer the coordinates to EM, do tomography to get high resolution, using either ultramicrotomy or focused ion beam milling to use this extent for tomography. And this field is separate, this volume microscopy. This has a lot of potential when you want to look at bigger objects like organs. I think I will end by thanking the Be Life grant, which funded the facility, the National Cryo facility. You can look in this webpage more, and I'll be happy to take questions.
Yes. Thank you, Dr. Vinod, for giving us such an enriching talk and in details, helping us understand that what are the differences between even electron microscopy and how it is useful and till what depth we can actually get an information from the cellular cell organelles. So if there are questions, you all can still go on putting here in the chat box, and Dr. Vinod will answer it. If not, then by thanking you, Dr. Vinod, to take out time from your busy schedule, and participating in a symposium. So thanks a lot. And we will move on to our next speaker. That's also on the applications of electron microscopy, of transmission electron microscopy in neurodegenerative diseases, by Dr. Rashmi Santosh Kumar. So she's a senior scientific assistant at the Electron Microscopy Common Research Facility at NIMHANS. And she's involved in the diagnostic electron microscopy as part of the clinical service at NIMHANS, and teaches ultrastructural pathology to postgraduate medical students. As a resource person, she was involved in several workshops conducted in electron microscopy and trained several students, faculties of various backgrounds. She's actively involved in guiding research scholars in thesis work involving electron microscopy. Her field of research is primarily myology, with special interest on inherited primary skeletal muscle disorders, protein myopathies, and autophagic vascular myopathies. Her key areas of research include also characterization of these SPAM and AVM using omics approach, development of model systems to dissect the disease etiology, and to explore therapeutic strategies to improve skeletal muscle functioning. She has several national and international publications to her credit, And with that, I will invite her to start out her presentation.
Thank you for the kind introduction, Dr. Indrani, and also thank you for inviting me to be part of this symposium. Good afternoon, one and all. After a wonderful talk of Dr. Vinod, I think some of these slides of the basic principles would be a little repetition. Nevertheless, I'll try to move on quickly on those parts. So I work as a senior scientific assistant in the electron microscopy facility at NIMHANS. So this is a common facility both for research as well as for diagnosis being the priority here. The samples that we receive here is basically from NIMHANS as well as from outside NIMHANS from various hospitals all over the country. And for research as well, students from NIMHANS utilize this facility for their thesis work, as well as students from various parts of the country, not only working in medicinal field, but also from various veterinary institutes or agricultural institutes. They use this facility for their thesis work. So I'll start off with my presentation. Basically, the presentation is divided into smaller subtitles. The introduction, as Dr. Vinod has already introduced, but still I'll just give a brief introduction and the principle of TEM and how we process samples here. Basically, what Dr. Vinod spoke was about the single particle, whereas here I would be concentrating more on the tissue specimens, and then how the sectioning and staining is taken care, and then we finally how the interpret these samples. So that will be the utility part of the TM. So this is the microscope that we have. It's a GEM 1400 plus Geol make from Japan. So the basic questions that we try to address is using EM is basically to look at the ultrastructure or the subcellular normal structures and what are the deviations or the alterations in these samples. And when do we use it is when we when we do not get any answers from the light microscopy. Or rather, coming from a pathology background, it tells that if we want to see something that's really abnormal. So this basically tells you that what is the limitation of our eye, that is the level at which we see, that is up to around 100 microns. Whereas when we compare that to the light microscope, we can see something up to around 1 micron thick sections. And what we actually want to see is in the subcellular level or at the atomic level, and which is possible only using an electron microscope. So that is like what viewing viral particles or macromolecules at the atomic level. So there are various kinds of electron microscope, the basic ones being the transmission and the scanning electron microscope. In addition, a few modifications like scanning transmission electron microscope, high resolution TEM, and low voltage electron microscope, where the voltage is between 5 to 25 kilovolts. And then cryo-EM, which I'm not going to touch upon, it's already dealt. So, and the correlative light and electron microscope. So today's presentation would focus basically on transmission electron microscope. So the history goes back to 1936 when the first electron microscope was invented or developed by the German physicist Ernst Röska, and along with his partner, who's an electrical engineer, that is Max Knoll. And for their discovery, they have got the Nobel Prize. And the main intention of using an electron microscope is because of its resolution. So basically, the resolution is defined as the ability to distinguish closely spaced points as two separate points. And this is dependent on the wavelength and the voltage. Wavelength, what I mean is the wavelength of the electrons. And this wavelength is variable based on the voltage that is supplied, which means if you subject it to higher voltage, then you will be having a shorter wavelength, and which is what we actually use in transmission electron microscope. This is the basic compound microscope which we all use day to day, and where we have the source at the bottom and the objective, and the eyepiece at the top. So just an inverted image of this is the TEM, where you have source on the top and the set of condenser lenses, and you have a screen which is fluorescent, where you can actually see the image. So when we take the factors that the comparison between the light and EM, you see that the source of illumination is photons versus the EM, where you use it as electrons. And the wavelength is in the visible range around 400 to 800 nanometers, whereas for EM around 100 kilovolts, you can see the nanometer range is around 0.0037. So the lens what we use is glass lenses. Obviously for electrons being the source, we have to use electromagnetic lenses. So the instrument is built for electromagnetic lenses. And the medium is usually air or oil in light microscope, whereas EM, it is the entire instrument has to be under high vacuum. And the magnification is really high. You can go beyond 1.2 lakh times. And the resolving power is around 0.2 nanometers. And the image that is formed is visualized on a fluorescent screen. And that is because electrons cannot be visualized through the naked eye. You cannot perceive it through your naked eye, and therefore it has to hit something that you can visualize, and that is the fluorescent screen, which emits light. So I will not get into the details of the working, but just to mention that the system has, the EM has three systems, that is illumination, imaging, and the image translating system. In addition, it also requires some ancillary equipments like high voltage power supply, vacuum pumps to maintain high vacuum in the entire instrument, and chiller to absorb the heat that is dissipated because of the high voltage that is used. And this chiller circulates water that is around 18 to 20 degrees centigrade, and an air compressor to maintain the humidity in the room. So looking at the principle, we have the electron gun. Here, the electron gun in a transmission electron microscope is either a tungsten or a lanthanum hexaboride, which is heated at a very high voltage. And we operate instrument here at NIMHANS around 80 kilovolts. And once the filament is heated at 80 kilovolts, it liberates a lot of electrons. It's called as electron cloud. And once that cloud is formed, the charge difference between the cathode, that is the filament, and the anode propels down these electrons, and focus is done by these condenser lenses as an electron beam. So ideally, we require a beam that has to fall onto the specimen. So the specimen is placed below the condenser lenses, and the image that is formed is magnified and projected. So magnified by the objective and the intermediate projectile lenses, and then the image is formed in a fluorescent screen. In a GEOL instrument, the camera is present below the screen, and therefore the screen is moved whenever the image has to be captured. So, and this is using a CCD camera, and the camera that we have is from Gatan. So basically, Dr. Vinod has already told that. Just to repeat, it is SEM versus TEM. SEM gives you only the topographical image, that is by capturing the secondary electrons that are eliminated from the surface of the sample. Whereas TEM, the electrons are transmitted through the sample, and the image that is captured is what we see as the electron micrographs. So it can be any kind of sample that you can process for TEM. It can be tissue samples or microorganisms, cultured cells, or any biomolecules. And the basic goal of processing the samples would be to preserve their architecture, preserve the native state of the sample, and to avoid artifacts. Basically, this is a very big challenge, because whatever images that you're going to see are going to be just white and black, and it is very important that no manual artifacts, and when you're handling the tissue, you're not going to introduce any artifacts manually. And you have to make the specimen stable under EM, because you're using high energy beams. And also we have to impart contrast to the specimen, because biological samples are not electron dense, and therefore electron density has to be increased by using heavy metals for staining. So basically the buffer that we use is very important. The tissue that we fix is, the fixative that is used is glyceraldehyde. That's the basic fixative, and sometimes a combination of fixative is used with glyceraldehyde and paraformaldehyde of varying percentage depending on the tissue, the hardness of the softness of the tissue. The pH of the fixative should be around 7.2 to 7.4 with a molarity of 0.1 to 0.2 molar. What we use at NIMANS right now is the sodium cacodylate buffer of 0.1 molar pH. Variation in the osmolarity or the temperature or duration of fixation can obviously affect the subcellular structures. There are two ways, the very basic ways of fixing. Either it is through perfusion, you perfuse the animal with the fixative and then dissect out the tissue, or you dissect out the tissue and immerse it in the fixative. Just a brief way how we process samples. It's initially primary fixation with the glutaraldehyde, which basically fixes all the protein moieties, which cross-links these amino groups. As I told, glutaraldehyde or Karnofsky's fixative can be used. Then it is post-fixation using osmium tetroxide and the series of dehydration with the graded series of alcohol. Then the clearing step with propylene oxide and then infiltration and embedded in the pure resin. So the resin here, what we use is called araldite, and polymerization is done at 60 degrees for 48 hours. So basically the dehydration step here is done in order to remove the water molecules, because the resin that we are going to use will be a hydrophobic resin. So when I'm talking about resin, it will be like there are two kinds of resin that are water miscible or water immiscible. So water immiscibles are the hydrophobic resins like araldite, spur, or ripon, whereas water miscible are the LR white and the LR gold. So these water miscible resins are the resins that are usually used for immunoelectron microscopy. And this is how the final embedding appears. Once the tissue is in osmium, it totally turns black, and we give an EM number identification to that sample, and it is embedded and it is allowed for polymerization at 60 degrees. Once after it's polymerized, then now each individual bits are ready to be sectioned, and the sectioning is called as ultramicrotomy, where you use to cut sections, you use glass knives or the diamond knives, and these are the glass knives. These are the makers which are used for glass knife. And this is how we are going to expose each of these blocks. You trim the extra tissue, and I mean the araldite, and this is a copper grid which is just 3.05 mm in diameter. We trim it to give it a trapezium shape so that you get serial sections like this when it floats on water. And these sections are collected on these grids, and then they are stained. So this is an ultra microtome which is used. It's a specialized microtome where it is used to collect these sections. Initially, a survey section or a semithin section is collected, which is around 0.5 to 1 micron in thickness. And this is transferred onto the glass slide to basically look for the area of interest, what you want to really observe, because there is a lot to see on EM. So you have to restrict yourself to what you want to exactly see, the specific area that you want to see. And then stain it with either 1% toluidine blue, which is made along with 1% sodium borate. So once the sections are identified, the area is identified on semithin sections, we collect the ultra-thin sections on the copper grid. So basically, the ultra-thin sections depends on the thickness of the section. And this is because of the interference. When the sections float on water, there is interference color based on the thickness. We usually prefer to take around 60 nanometer to 70 nanometer thick sections, which is around the silver color sections, or the silver to golden color sections. The thicker the section, the penetration of electron beam is going to be reduced. So as I told previously, we stain these biological samples basically with uranyl salts or lead salts to enhance or give them a better contrast when you observe under EM. So this is a goniometer or the sample holder, and this is a slot where this 3 mm grid is introduced into, and it will be introduced into the electron microscope. So in addition to the EM, the regular EM, there can be certain modifications of these techniques, like using light microscopic techniques, including histochemistry or immunohistochemistry or autoradiography, as well as techniques that are other than the regular staining. You can also use negative staining or a freeze fracture or shadow casting. So coming now to the utility, basically, as I told you, initially we are into diagnosis. So we routinely get skin, muscle, and nerve to diagnose for various neurometabolic disorders or the storage disorders. It could be either neuromuscular or It could be peripheral tissues like nerve. So the basic idea here we do is for diagnosis. The second thing is to confirm the diagnosis, because there are certain conditions wherein we do stains, wherein you preliminary get some information, and that information, if you want to confirm further, we use EM for that. In some conditions, we use it for complementing, like neurodegenerative disorders, for example, Alzheimer's disease. And in the most widely used application is in research. It could be looking at the normal structures, or it could be the abnormal or pathological alterations, or looking at the cell culture studies or specific localization of any particular protein or any other macromolecules. So before interpretation, we need to actually know how a normal looks, and then we'll be able to actually justify as to what are the abnormalities or the cellular changes from the normal picture. So this is just a simple cartoon showing the eukaryotic cell. There are various subcellular structures, and each of these organelles have their own appearance, a characteristic appearance, and that comes with their membrane structures. So for example, nucleus is the most or the biggest organelle within a given cell, which has a double membrane, whereas there are various other organelles which are made up of only single membrane structures. So this is just a portion of the grid. It is like just one portion, and this is 3 mm copper grid, onto which, if you see the silver ribbon, is the sections that are collected onto this grid. So when we start looking or interpreting the tissue, the first thing we look at how well it is preserved to avoid the changes that is introduced during the processing. So this is just an example of a section through the liver. You have the hepatocytes here, and this just one portion enlarged is what you see on an EM, the entire cell. So this is the largest organelle is the nucleus that we are seeing. So in the same way, we have to know what are the basic structures. So some of the cells have a additional membrane. It is the basal lamina or the basement membrane. For example, in case of muscle, it has an extra membrane outside the plasma membrane, which separates the myofiber from that of the extracellular space. And so plasma membrane, this is the high magnification showing a plasma membrane of 2 adjacent cells. Other than myofiber, the basement membranes can also be seen in the nerve, myelinated and the unmyelinated nerve twigs, or even in the endothelial cells. They all have a basement membrane in addition to the plasma membrane. So when we look at the nucleus, we can see that there is a nuclear membrane, and this is the nucleolus, but we are not able to see at a very low magnification the double membrane of the nucleus. But what we can see is that, yes, there's a nucleus with the nucleolus and the chromatin material. So the chromatin, if it is heterochromatin, because it's highly condensed, it becomes more electron dense. So what you're seeing on the right is the mitochondria, a single mitochondria, where you can see the outer membrane and the inner membrane, which is continuous with the crystal that is thrown into folds. So a few more images of the normal structures, especially the RERs, which you can see these are the granular structures to make that is the ribosomes, and hence they are called the rough endoplasmic reticulum, which is very well seen on this image on the right as well. And the Golgi complex, which usually in the form of tubules, they are in stacks, and they usually bud out vesicles. They are secretory in nature, and therefore you see them budding out vesicles. And this is an image of smooth reticulum. It's a huge network of the reticulum seen here. And just in one image to show the different organelles, in addition, you can also have these lysosomes. Lysosomes appear very electron dense. They're the suicide bags. So they have this electron density because they have those degraded material accumulated in them. So in addition, some more normal structures like the desmosomes, these are the important junctions that you see, basically seen in all these layers of epithelial cells, which can be present either on the basolateral surfaces, lateral surfaces, or the basal surfaces. So this is just a portion of a glandular epithelial cells where you can see numerous desmosomes. On a very high magnification, you will be able to appreciate that there are the inner dense plaque, and then the outer dense plaque, and then is the plasma membrane and the intercellular space. So this is on a very high magnification. You will be able to appreciate the desmosomes. So the abnormalities in hemidesmosomes can result in separation of these cells that are joined together. And one of those conditions due to that is due to the separation causes these bullous lesions called the epidermolysis bullosa. This is an example of a gap junction. It's a normal feature that is seen in the ependymal cells. So these are the characteristic features that helps you in identifying these cells. So the most interesting part here is the axodendritic synapses. So these are the boutins with the neurotransmitter carrying vesicles, and this is the postsynaptic density. And this is within a tissue. So when students isolate it, they have various goals to use this for. One is either to look at the synaptic content or the proteins that are present in the synaptosomes, profiling of these proteins, and to characterize this preparation, they use this electron microscope facility. So the other normal features are like the centrioles, which on TS shows that they are centrioles with triplets. They have triplet tubules basically made of the microtubules. In contrast, when we look at the cilia, they have a 9+2 arrangement. This cartoon shows that they are the tubules, the A and the B tubule, and they have the outer dynein and the inner dynein arms and the radial spoke, which all work together in the movement of the cilia. So if there is any defects in this, for example, when you look at this picture, you are able to see that there are only the microtubule doublets, but there is absence of these dynein arms and even the radial spoke. So these kind of cilia are usually seen in patients who suffer from primary ciliary dyskinesia, wherein the movements are hindered. And usually you can see these from samples like a nasal biopsy or a nasal brushings. So various clinical features are associated with primary ciliary dyskinesia. But what you see on EM is you have to look for those dynein arms and the radial spokes if they are defective. And you have to also see how many such cilias or the axonemes show these changes before you interpret whether it is a condition of primary ciliary dyskinesia. So as I showed the normal structures, we also try to see the irregularities in the these basement membranes, wherein this is a capillary which shows a thickened basement membrane. So thickened basement membrane could be a normal feature in diabetic patients. But when you're interpreting, you have to be careful and taking out the clinical features and comparing them with what condition the patient is suffering from, and then we have to actually decipher. So in addition, in case of skeletal muscle disorder, where it is called spinal muscular atrophy, you can see that these muscle fibers have got atrophied and there is redundant lamina, that is the basement membrane is just thrown into folds. So these are some of the abnormalities you see in the basement membrane. This is a normal nucleus, and there can be some abnormalities even in the nucleus, like this, which shows there is accumulation of intranuclear filaments due to some conditions like the actin or actin myopathies, wherein the actin mutations can result in accumulation of such filaments within the nucleus. These are some of the abnormalities of rough endoplasmic reticulum. Usually they are the tubular structures. You can see some dilation, some in cases of infection, as well as this should be not confused with the artifactual change due to a hypomolar or the molarity, if the change in molarity of the buffer, you can also have such conditions. But this is a condition wherein you are seeing there is dilation due to an infection. And this is the infection with the JE virus where the viral particles are seen within the endoplasmic reticulum. So usually the normal Golgi complex is in stacks with budding out vesicles. Due to some pathological changes, the Golgi can undergo fragmentation and there are just only vesicles seen there. So the most fascinating is the mitochondrial changes. There are a group of mitochondrial myopathies, and they can show varied changes in the cristae pattern. So this is like electron dense, and they don't have the routine or the normal pattern of folds, but have changed the cristae alteration, like the pattern of cristae here. This is the lamella or the concentric pattern. This is sort of ballooning of the cristae. And here we have vacuole formation, some electron dense structures accumulated, and also a lamellar cristae within the mitochondria. In addition, mitochondria can also have some inclusions, and they are called as paracrystalline inclusions. These inclusions are basically the content is nothing but the creatine kinase, which crystallizes here in between the folds of the cristae. And that's why it takes that proper arrangement, like a car parked in a parking lot, and hence the name parking lot inclusions. These are type 1 kind of inclusions, and they are present between the folds of the cristae. Whereas type 2 paracrystalline inclusions, they are just present between the outer and the inner mitochondrial membrane as just one inclusion, and not like present in the folds of the cristae. So these were the paracrystalline and the abnormality seen in mitochondria. Now we'll just move on to the peripheral tissues that we usually get for diagnosis at EM. The first one is the skin biopsy. What we see here is on a light microscope. This is the epidermis region and the dermis with the dermal collagen. And when you look at the deeper dermis, you can find these glands. So basically, we get skin biopsies for the diagnosis of various neurometabolic disorders. Initially, the diagnosis was based on the biopsies taken from either the brain or the rectal samples. But now, skin biopsy is the most convenient, comparatively less invasive technique, and a peripheral tissue which can help you in diagnosis of these conditions. So on semithin, these are the sweat glands, the apocrine and the eccrine glands. So on EM, this is how you see on a low magnification, the entire structure of the gland with some secretory granules, the lumen, and there is the, just next to the lumen is covered by the epithelial cell layer, and the outer is the myoepithelial cell layer. And similar to that in an apocrine gland, how we make out the difference is the apocrine glands have a larger lumen compared to the eccrine glands. So as I told, we get samples for diagnosis of various neurometabolic disorders, and I've taken just one example here about the neuronal ceroid lipofuscinosis. So basically, the diagnosis can be done using genetic studies. In case of using electron microscopy, there is characteristic feature when we look for on EM, and it is based on the age at onset, the clinical features also. So when we look for NCL cases, they are classified into 4 different types based on the ultrastructural studies. First classification is when the age at onset is between 0 to 2 years. It's called infantile NCL, where you see these deposits in the lysosomes. Basically, these are lysosomal storage disorders, and therefore they are seen as granular osmophilic deposits. In the next classification, that's late infantile NCL, it is 2 to 4 years. They are curvy linear bodies. In juvenile NCL, where it is 4 to 9 years, you see them as fingerprint bodies. And the fourth one is an adult NCL. We usually have not received any samples of adult NCL, or where we haven't found any inclusions rather. So this is a list of genes and proteins. Now that you can also have the genetics done initially before doing a biopsy. In case if there is no information, or if you don't get any information from genetics, still the gold standard would be using EM to identify these inclusions. So this is on a low magnification, showing an eccrine gland. That is a portion of the eccrine gland. This is the lumen. And here is the myoepithelial cell. So at this magnification, you'll not be able to appreciate the inclusions, but when you go on a very high magnification, this is how the granular osmophilic deposits are seen. So this is the example from the infantile NCL. When we move on to the late infantile NCL, you see that clearly there is a membrane, and these inclusions are present within these membrane-bound organelles, that is the lysosomes, and they are called as curvilinear because they have an inverted C-shaped structure. So if the organelle is totally full with it, the membrane will not be very evident, or else you can sometimes see this structures. So the next one is the proper fingerprint inclusions, which is nothing but like our fingerprints, and therefore the name fingerprint inclusions, which is seen in the juvenile kind of an NCL. Sometimes you can have in juvenile NCL both the curvilinear and the fingerprint inclusions. So these are the curvilinear, which has a slightly lamellar pattern that is seen, and this is well within the lysosome. So these inclusions can be seen in any cell type other than glands. Also, it can be seen in the endothelial cells of the blood vessel. It can be seen in the Schwann cells, can also be seen in the adipocytes, as well as even in the nerve twigs that are present. So the next sample or the frequently received sample is the skeletal muscle for looking at the neuromuscular disorders, especially where skeletal muscle is the primarily affected tissue. So this is on a light microscopy, wherein you see the section showing fascicles. There are at least 2 to 3 fascicles. And on EM, you'll be seeing this just portion, which is portion of, or the junction of 3 myofibers. So each fiber or each cell here is called as a myofiber. So each one of this is a myofiber. And in between, what you're seeing is the interstitium, where you have the fibroblasts. So these myofibers, on a higher magnification, you can see the myofibrils. So each fascicle is made up of multiple myofibers. Within each myofiber, you have myofibrils, and each of this fibril on a higher magnification shows the hexagonal arrangement of the actin and the myosin, that is the thick and the thin filament arrangement. So on light microscopy with Toledan blue stain, this is how you see as longitudinal section with the striation pattern. And on EM, you can appreciate the striations as the dark and the light bands, the isotropic and the anisotropic bands. On a higher magnification further, you can see each individual sarcomere with the various bands within the sarcomere. So this is nothing but the structural unit of the sarcomere is the structural and the functional unit of the muscle. So I'll just present one here, a case of desminopathy. Desmin, as all of us know, is a protein that is expressed very early at in the embryonic stages itself. So here is a case wherein desmin is totally absent in the tissue. And it is also seen that desmin is responsible for maintaining the architecture of the myofiber, and it also connects the nucleus to the membrane. So, and it also positions various cell organelles within the muscle fiber. So basically, if there is total absence of desmin, you can see in this inset, this is a HE stain portion of just one muscle fiber here, showing accumulation of nuclei, which is stained with hematoxylin. There is aggregation of nuclei in the subsarcolemmal region of the muscle fiber. And what we see on EM is this sort of an aggregation wherein the structure is totally lost and they are all moved towards the subsarcolemmal region and they are called aggregated. They're sort of indentations. Usually the muscle nuclei is oval to round and they don't have such degree of indentations. So this is one abnormality in a case where there is total absence of desmin. In addition, as I showed in the previous structure, this sort of an appearance of the sarcomere, the registry, is totally lost and you have lot of aggregation of the Z-band material here. So here are some of the fibrils still having the integrity maintained, whereas here it's totally lost and it's getting the entire filamentous structure is sort of distorted and they are all aggregated in the subsarcolemmal region. So now moving on to a peripheral nerve tissue. So in peripheral nerve tissue, this is on semithin section. What you see here in this circle here is the myelinated fibers. All these blue rings are the myelinated fibers, and this is just portion of a TS of the peripheral nerve tissue. What remains unstained is the unmyelinated nerve fibers. So this is on a Toledan blue staining. So when we look at just one myelinated nerve fiber, you see that this is an entire Schwann cell nucleus, and within the cytoplasm, you can see the myelinated axon. So within the axon on a higher magnification, you can see that there are various proteins in the form of either microtubules or filaments that are cut in transverse section, which appear as just black dots. In addition, there are various organelles, that is the mitochondria. So this is within the axoplasm, and you can see the beautiful lamellar pattern of this myelin ring, which is formed from the plasma membrane, which goes around to form the intraperiod lines, the dense lines, and the lesser dense lines, which is appreciated only on electron microscopy. So the unmyelinated nerve fibers, when we compare that to the myelinated fibers, here there's only one axon that is surrounded by the basement membrane. Whereas in case of unmyelinated, you see that one basement membrane is seen to hold multiple or cover multiple axons. So moving on to a condition called Fabry's disease, which is an X-linked disorder and which can be diagnosed using either a skin biopsy, which should have either nerve and the blood vessels on a semetin, which you have to ensure that, and then or else you can also see it on a peripheral nerve tissue. So this is a condition called Fabry's disease. It's an X-linked disorder due to the deficiency of a lysosomal enzyme, that is alpha-galactosidase A, which is encoded by the GLA gene, which results in the accumulation of these glycosphingolipids, especially the globotriaosylceramide ceramide. So ultrastructurally, you see them as electron-dense, concentrically tightly packed lamellar structures. And you preferentially see these inclusions in the endothelial and the vascular smooth muscle cells. It could be even the neurons, perineural cells, but you cannot see these inclusions in the Schwann cell cytoplasm. So this helps us in differentiating that from a metachromatic leukodystrophy as compared to the Fabry's disease. So basically, these kind of accumulations result in the various the symptoms that you see in the patient. And as I told, it's an X-linked disorder. The male patients are seen to be highly affected, and the female patients are usually the heterozygous carriers. So as I mentioned, what kind of inclusions we see is these zebra body inclusions. So this is a peripheral nerve biopsy. On a tolidin blue section itself, sometimes you can pick up that there are certain the intensely stained structures, which in the perineural cells appear as lamellated structures, and on a very high magnification, you will be able to appreciate this zebra pattern. You have concentric arrangement of the dark and the light pattern, giving it the name zebra body inclusions. So next, we move on to a condition wherein you use EM, which helps in not only looking at the structure, but ideally by basic staining techniques, you can identify these. That is, in case of Alzheimer's disease, you see these neurofibrillary tangles in the pyramidal cells of the neurons, which have this flame shape. And this can be seen even on the Bodian stain. This is the Bodian stain showing a neurofibrillary tangle in the pyramidal cells. If you have to appreciate or look at the change, how it appears, these tangles, this is on a low mag, and on a high mag, you can see these are the paired helical filaments in the neurons. So this is a case wherein you can use EM as complementary for what you have seen on the basic histochemistry technique. The other condition is looking for the cytoplasmic inclusions. Here they are the Negri bodies. This is caused due to the infection with rabies virus. Rabies virus are these particles which have a characteristic pattern. They are bullet-shaped viral particles, one of the larger viruses. They have an electron-dense core surrounded by something how we see in a corona with that crown kind of an appearance with an helical structure, the helical core structure. And this on the Negri body on EM shows all the viral particles sort of accumulated in the core, and the filaments are seen at the periphery of the inclusion. So now we move on to some of the examples that where students have used this for research. One such example is for characterization of Mycobacterium tuberculosis, where the bacteria was isolated from various patients suffering from TB, CNS TB, and they have used the CSF to isolate it. And they have seen that the resistance to the mycobacterium for various drugs is because of the cell wall. So the cell wall component is the one that actually responsible for causing the pathology. Now, if the cell wall thickness is increased, they are not permeable to the anti-mycobacterial drugs. So these cell wall thickness is what is measured, and it is seen that these resistant bacteria have a maximum difference of around 26.48%. That is, the thickness is more in these resistant compared to the sensitive isolates, and also when compared to those which are sensitive to either just one or two drugs. So when they are multi-drug resistant bacteria, they have highly thick walls, and these thick walls are very well appreciated on higher magnification, which has 3 layers and which prevents the entry of these drugs into the cells, and therefore they are free to cause the pathology. So another important technique that is used is immunoelectron microscopy by using staining or antibodies to particular proteins and to localize these proteins. This is one such publication from Dr. Govindaraj Padmanabhan, and along with a collaboration with Dr. Sagar, where they have shown that the delta aminoleuvelinic acid dehydratase, this is an enzyme in the heme biosynthetic pathway. This enzyme is present in the RBC because it contains hemoglobin. There is heme biosynthesis happening here. So in the heme biosynthetic pathway, these enzymes are...
She's not here. Maybe she's talking with sir. It's there 10 minutes before. Yeah, she's here.
Yeah, sorry for the interruption. So the other technique that is used is immunoelectron microscopy, where this technique was used to localize the enzyme that is delta aminoleuvelinic acid dehydratase in the RBCs. And it is also seen that this enzyme is utilized by the parasite for the de novo synthesis of heme. So, and that is localized to the apicoplast of the plasmodium parasite. So this is a method used for localization, and this technique helped them in identifying the localization. In addition, it also helps to target this region or these molecules for drugs, so that they can avoid or cause defect to the pathway, and hence their replication. Dr. Vinod also highlighted on another technique that is negative staining. This is a technique wherein you use pure samples like either viral particles in solution or proteins, where you use them to look at the structure, visualize the structure, and you can also measure these structures. So basically, the procedure for negative staining is like this. The grid is coated either with a foam wire or a colloidal or carbon coating, followed by using just a sample. A small volume of sample is sufficient to do this procedure. And then you stain them with uranyl acetate or phosphotungstic acid. You have to dry the grids and then visualize it under PM. So some of the examples are, one is that negatively stained the chick virus, that is the chikungunya virus. This was done by one of the student called Ayushman from Neurovirology Department of NIMHANS, where he used this virus for further looking at the pathological changes after infecting them with C2C12 cell lines, which is the myoblast cell lines, and the mosquito cell lines, that is C636, to look at the cytopathic effects. So basically, the chick virus, before infecting the cells, are for the appreciating their structure, the negative staining is used, where they have a spherical appearance with a electron lucent central core. The other important one is the use of exosomes, and you can characterize exosomes using this negative staining. And one such use is as a biomarker, wherein a student from neurochemistry has used this. Ms. Gitu has used this to study the protein concentrations, or the lysosomal associated membrane protein 2 in particular, where she has isolated exosomes from the plasma of patients and the controls, and to characterize or to confidently tell yes if this is the exosomes or not, the TEM facility was used, and the exosomes were characterized. And she has also seen that the proteins isolated from such exosomes from patient and AD in the comparison Alzheimer's patients showed that there is higher levels of lysosome associated membrane 2 complex, which shows that there is effective lysosomal activity, and there is aggregation of lot of proteins within the cells. So finally, coming to electron tomography, which I'm not going to stress more on because Dr. Vinod has already done a lot on this. So basically, it is just a technique till whatever now we have seen is all the EM graphs, which are white and black. But yes, you can make them look more colorful, make them look more better, as well as analyze them in a better way by doing electron tomography. You can use the same sections for electron tomography, where the section is tilted, where the grids are tilted at an angle of plus to minus 70 degree, and at a you lower them down by 1 degree and you get multiple images, and then you reconstruct them computationally to create the image. So basically, this is one such paper which shows the three-dimensional structure and organization of the cristae. And using this technique, they have tried to look at the points where the inner membrane or the cristae meets the outer membrane, which is a point where there can be the exchange of molecules from the cytoplasm into the mitochondria. So coming to the final slide, that is the take-home message. It is very important tool. It is an invaluable tool in the diagnosis as well as for research. The student should always be careful. You have to use a proper fixator, and the time of fixation is very important. It's a very crucial step. Everything depends on this particular step, and if there is anything, if there is any mistake done in the fixation, the interpretation is really tough. So, and always keep in mind that you need to have a control sample for comparison with your experimental or your test samples. Thank you. I would like to take this opportunity to thank my mentors and the entire electron microscopy team and their support for all the guidance that I've got from my mentors. Thank you so much.
Thank you so much, Rashmi, for this wonderful presentation. And indeed, we enjoyed all the beautiful images you have shown with respect to diagnostic and with respect to research, be it from the exosomes to the lysosomes to the mitochondria and the cristae and the Golgi apparatus. And it was really, we wished that it didn't end. We enjoyed every bit of it. So if there are any questions, please leave them in the chat box. There are some. So I would, yes. So Rashmi, you can go ahead and answer them.
So Ms. Divya, would you please give me some idea of microorganism TEM picture? Is there any separate procedure for it? How to avoid contamination in image? We don't have a scanning electron microscope. What we have is a TEM. So in case of microorganism, it depends as to what sort of an organism it is. Whether it has a really thick coat, you have to use a different resin, and slight changes in the protocol from fixation till the dehydration steps and the resin finally in which you embed. For example, you can use a spur resin and the dehydration as such, it could be in alcohol. I think this would be one of the ideas. In particular, you have anything like a particular organism. For example, I showed you a paper wherein Mycobacterium tuberculosis was used. In that paper, the spur resin is used for the processing. Give example, especially in bacterial sample. So that is the example that I can remember right now. You can write to me, and if you have any queries in particular that you want to visualize, and I can address it. It's the only question that they have.
Thank you so much, Rashmi. And if y'all have any questions later on, it's coming up when y'all are going through and thinking about any of the topic, you're most welcome to write back to the mail address of a symposium. We will try to get back to the concerned speakers and communicate back the answers or send you with their permission, their email addresses. So with that, the theme, the second day's second half or second session's theme also ends with the advances in microscopy. And it was a little jam-packed, I do understand, especially the first day we had too many presentations also along with the speaker presentations. But you know, at the end of the day, we are over here to get condensed amount of knowledge and get the experience from the experts and in the field of imaging, be it from the hardware point of view or be it from the imaging application perspectives. So the more we get, the better it is. So with that, I will start, just take a few more minutes from you people, all of you, to give the vote of thanks. And before that, I would also like to announce that Ms. Reena Bharti, I don't know whether you're present here. Yes, I can see you. So you have, congratulations, you've got the prize for the oral presentation among the students category. So we will send you the certificate through mail. And so I would like to start with a vote of thanks. First and foremost, I would give my heartfelt thanks to the patrons of the symposium, that is our director, the NIMHANS director, sir, Dr. Guru Raj, and the head of the Department of Biophysics, Professor Padmanabhan, for extending their help, their cooperation, and giving us the encouragement to go ahead with this symposium. This is first of a kind that we have held this time. As I said, we usually do the hands-on workshop with theory and practical classes. And due to the pandemic and lockdown, we had to change and shift it, but we wanted the discussion and the science going, and hence we conducted this. We learned a lot also from our experience over here and hope to make it better eventually. I would also like to extend my thanks to our registrar, sir, Dr. Shankar Narayan Rao, and to the head of Telemedicine Center, Dr. Suresh Padamat. Without his help, it wouldn't have been possible for us to conduct this online symposium with least amount of tension and be running it as smoothly as it is. Without his team, it wouldn't have been possible. So Naveen and Adarsh from the technical support, and been staying extra hours with us. That was really nice of you all, and we appreciate it from the bottom of our hearts. And without the speakers, this wouldn't have been so exciting, and this wouldn't have been the thing what we wanted it to be. So my heartfelt thanks to all the speakers for taking out time. And I know many of the speakers were from outside India, and they were very unearthly hours that they had to log in and get in for the talks. And for some of the speakers, even for the first day, few minutes delay, there was delay in presentation, but it was amazing that I got a response from all of them, and they were always on point, and all of you all wanted to be there also for the other sessions, so that enriching the symposium together. And all of this wouldn't have been possible also without all of you, the participants, being present on time, showing the enthusiasm, and being there for all the sessions, be it with your questions or be it listening. At the end of the day, if I'm doing or we are doing the symposium from here, it is mainly that even one or two learning experience you take back to your research, to your bedside, and you apply it. So tomorrow when you go ahead with looking into your cells under the microscope or looking into your in vivo model and going ahead for any study of transplantation, you have certain key points to remember or to jot it down when you are designing your experiment. That would be the fulfillment which we will get through our symposium. So thank you to all the participants. I would also like to extend my thanks to the sponsor, Eppendorf, and I thank the Department of Biophysics. And along with that, I have a very, I don't know really in what words I should say, thanks to my organizing committee, that's my students. They have been running around and making timely efforts and organizing this and making the schedule and everything well smoothly so that I can come and just coordinate the talks. So thanks a lot to my team for doing this backup job so well. And I have a request to all of you. Please kindly fill in and submit the feedback forms which has been sent to all of you through email and try to provide it by today. Because if you all finish off sending us the feedback forms, we will be initiating to send the certificates one by one. And by the end of the weekend, we are hoping to send the certificates to all of you. So feedback forms, why we are saying is important because it is not only to know, because it's an online thing, so we are missing that one-to-one interaction and experience because end of the day we are still human beings. So through the feedback forms we'll get that feel at least, and also to say that we will be able to improve on the topics which we have missed on, and might be in our next symposium we will try to include those topics from the perspective of imaging and stem cells. And also, we will be able to get to look into a third person's perspective that what we need to improve on. So please don't ignore it. And it's a sincere request to cooperate with us and give us our feedback form, because we also want to communicate it to ICMR. They are being a big support for us and allowed us and gave us the permission to be it under the aegis of ICMR. So for that, I again thank from the bottom of my heart, especially Dr. Geeta Jotwani and her team for being with us for this, be it for the workshop and be it now for the online symposium. So with that, I will conclude and I would say bye to all of you all. Best wishes for your career and stay safe. Keep social distancing still now. Thanks a lot.